Hello, everyone, and welcome back to Creation Myths. On May 2nd, I got to debate Dr. Casey Luskin from Discovery Institute on the Non-Sequitur Show on the topic of junk DNA, and that is what I'm going to show you right here. We talked about a whole bunch of different things, different parts of the genome, what they do or don't do, what they might do, and what they can or can't do. As you watch, the thing I want you to see is how Dr. Luskin's argument evolves, if you will, over the course of this debate. At the beginning, it's we have documented function for most of the genome. And by the end, it's, well, we don't have documented function for most of the genome, but the trend line says that we will, so it's reasonable to think that we will. My position, the whole time, is that we not only don't have function documented for most of the genome, but we've actually dug into what is present in most of the human genome at this point, found a bunch of it is functional, including regions that we didn't previously think were functional. And that stuff adds up to about 10%. And then a giant chunk of the rest of it actually can't do any of the documented functions according to the sequences that we find. Not only that, but they can't do some of the purported functions based on the sequences that are or are not present. So if you want to say the rest of that stuff is functional, you need to propose new functions for it and then document those. Now, Discovery Institute has already put up some responses to what I said here. As I record this, there's at least one article responding to a specific thing that I said during this debate. So I will have more on this as I kind of work through their stuff in response to this debate. But for now, let me just show you the debate. Please enjoy.
All right, welcome everyone to the Non-Sequitur Show. Tonight we have an absolutely incredible debate for you all. Something that's been a work in progress for quite some time, to say the least. But first, let me introduce my two guests here. I have Dr. Casey Luskin, who is a scientist and attorney with a PhD in geology from the University of Johannesburg and a law degree. He ain't messing around. He's got a little bit of qualifications for everything. From the University of San Diego. He holds a master's degree and a bachelor's degree in earth science from the University of California, San Diego. He works at the Discovery Institute as an associate director of the Center for Science and Culture, where he helps direct the ID 3.0 research program and assists and defends scientists, educators, and students who seek to study, research, and teach about scientific debate over evolution and intelligence design. And on the other side, we have Dr. Daniel Stern Cardinal, who is an evolutionary biologist with a PhD in genetics and microbiology, who is also known as Dr. Dan and has a YouTube channel by the name of Creation Myths. First, I really do want to thank both of our debaters tonight for being here in what I think has been nothing short of a major journey, so to speak, in regards to how much of the human genome is considered to be junk DNA and the functionality of that junk DNA in the human genome. Just to let you know, we've allotted for about 90 minutes to 2 hours for this debate, with both debaters having about 10 minutes for their opening statements, followed by open discussion. And then, if time permitting, hopefully we'll have a Q&A. So with that, welcome, gentlemen, to the Non-Sequitur Show. And you know what? How about we just dive in? Unless there's anything you want to say first. We've already decided that Casey was going to start the debate, but if there's anything you want to say prior to that, let me know now.
I think we're good. Let's do it.
Yeah. Okay.
Right,
Sounds good.
And welcome, audience. Hi, all you people watching out there as well. Let's welcome them. So Casey, you have the floor. Take it away and start us off.
And I think that all three of us are either sick or dealing with allergies right now. So we're going to sound really terrible tonight. We're going to be hacking and coughing. It's going to be a miserable night, but at least we have company together. So thank you so much, Steve, and the Non-Sequitur Show for hosting us. And thank you, Dr. Dan, for being willing to have this conversation. I'm going to sacrifice most of my opening statement to provide some basic framing for our audience rather than getting too much into the technical details right now. I think a lot of that will come out during the conversation. So let's start by asking, why are we having this conversation tonight? Why does it matter if the genome is largely functional? If there is any quote unquote junk in the genome, does that mean that we've refuted intelligent design? Or if there is some of the junk in the genome turns out to have function, does that mean that we've refuted evolution? Well, on both counts, I'd say no, and I don't think I've ever said otherwise. However, it does matter whether our genome is largely junk because ID and evolution have made very different predictions on this point. So my 4 main points tonight are going to be as follows. And of course, we'll drill down into a lot more detail. 1, the best evidence shows that our genomes are largely functional and that junk DNA is the exception to the rule of mass functionality in our genome. In fact, we've seen a paradigm shift over the last 10 to 15 years away from the idea of junk DNA. Number two, intelligent design as a scientific theory predicted this evidence and predicted this paradigm shift. Number three, evolutionary biology, by and large, did not predict this evidence and even opposed this paradigm shift. In fact, I think as we're probably going to see in this conversation, evolutionists today are still strongly pushing the idea that our genome is largely junk. And four, this means that evolution has served and maybe even still serves as a science stopper, holding back science from discovering the functions for junk DNA, while ID has led to fruitful scientific predictions. At least that's what I'm going to maintain. So again, we're going to dive deep into this today, but I think it's good to start with some points of agreement. I fully agree with Dr. Dan, and I'm assuming this is what you think, Dr. Dan, so forgive me if I'm misrepresenting you, but I thought these were reasonable things, that there's still a lot we don't know about the genome. And I agree with you that probably some of it is going to turn out to be junk. I also agree with you that there's a lot of transcription going on where we still don't know what it's for. Some of it may even be quote unquote stochastic. But I certainly agree that there's a lot we don't know and that science will reveal much to us in the coming years. And I also need to say one thing to Dan before we go any further, and that is in one of your videos, Dan, you said that one of the photos that we put up of you on an Evolution News post was not a very flattering photo. And I want to say that I had nothing to do with that. We have somebody who edits our website that puts up, banner images for all of our posts, and they put it up. Honestly, I hadn't even really noticed it till I watched your video, but I asked them to put up a different photo of you because you're a good looking guy and we want the world to know that. And I asked them to put up a much more flattering photo of you. So if you go back to any of the posts we have on you, it's going to use a much more debonair photograph of you there. So it wasn't hard to find a good photo of you, by the way. Anyway, so a lot has happened in the last 10 to 15 years. And I think it strongly tells us where the trend line of the data is going. A hallmark of a good scientific theory is whether it makes good and successful scientific predictions. So what are the different predictions that ID and evolution have made? So while function for junk DNA may not strictly speaking refute evolution, the discovery of widespread functionality for non-coding or junk DNA represents a failed prediction for evolution. And while function for junk DNA doesn't necessarily prove intelligent design, the evidence of mass functionality for junk DNA represents a spectacularly successful prediction of ID. Now, as I mentioned, for decades, the evolutionary paradigm predicted and maintained in the absence of clear evidence, basically, I think, inferring in the absence of evidence that our genome was largely junk. If you want quotes on this, you can go to evolutionnews.org right now and find the post at the top titled Junk DNA from Three Perspectives, Some Key Quotes. And you can find lots of quotes from evolutionists who are asserting that the genome is largely junk. And this view flows naturally out of their paradigm. Their paradigm says that ultimately we are the result of nothing more than random mutation and blind natural selection, plus genetic drift and a suite of other blind mechanisms, neutral evolution, et cetera. So over time, evolutionists would propose that various classes of random mutational events, from whole genome duplications to individual gene duplications to viral insertions to parasitic, selfish DNA elements spreading copies of themselves through the genome, to random point mutations, that these have filled our genomes with junk and what is effectively evolutionary debris. And they would say that no designer planned our genome, and the genome is inefficiently bloated with things that do nothing to enhance our survival. So exactly how much junk do evolutionists say is in the genome? Well, you get different numbers depending on who you ask. But to give a few examples, in the early 1970s, late 60s, Susumu Ono, a very famous Japanese biologist who is credited by some as coining the term junk DNA, and he was an evolutionary biologist, he proposed that perhaps 90% of mammalian genomes are junk. Francis Collins said about 45% of our genome is at least, this repetitive DNA that isn't doing anything for us. Dan Graur, a very well-known evolutionary scientist at the University of Houston and quite a colorful character, says around 85+% of our genome is junk. Larry Moran, also an internet character that many folks will know of. He wrote a book last year. He's a professor of biochemistry at the University of Toronto. Said that somewhere around 90% of our genome is junk DNA. It's the subtitle of the book. Dawkins once said in I believe it was The Greatest Show on Earth, that upwards of 95% of our genome is junk. So what does ID say? Well, to explain this, I want to share some of my own personal story here. I think it's always nice to tell personal stories. In the late 90s and early 2000s, I was an undergraduate student at the University of California, San Diego. And during my junior year, some friends and I started a student club called the Intelligent Design and Evolution Awareness Club, or IDEA Club for short, to discuss intelligent design. And at that time, we would openly invite, ID skeptics and critics and evolutionists to come and have friendly conversations like us, just like what we're doing tonight. So I've really enjoyed doing this sort of thing. And we were constantly hit over the head with the argument that the genome is largely junk DNA. And we were told that no intelligent designer would ever put so much junk into the genome. And this was good evidence that the genome was not designed. Now, keep in mind that this was before the Human Genome Project came out around 2003, where it was fairly well established that maybe only 1% to 2% of our genome actually encodes proteins. But even at this point, people were pretty sure that, a very, very high percentage of our genome does not encode proteins. And they basically just assumed that non-protein coding DNA was largely junk. And this was an absolute defeater of intelligent design. But there was a problem with this argument back then, and it's pretty much the exact same problem that I believe that many evolutionists face today. And that is that this claim was made out of ignorance. It was based upon an absence of knowledge of what the non-protein coding DNA is doing, and the assumption that if we didn't know what it was doing, then it must be useless junk. Francis Collins later called this kind of thinking hubris. So what would we from the ID camp say in response? Well, we would say that if the genome really is largely junk DNA, then intelligent design would not predict this. We would say that they've actually sort of framed the issue in a fair manner. But we would also say, especially at that time, that we certainly did not know enough about the genome to say that non-protein coding DNA was largely useless genetic junk. So rather than assuming that it was junk, we would say, let's take a wait and see approach and let the evidence decide. And this was before there was hardly anything known about what the non-coding DNA was doing. Again, this was the late 90s, early 2000s. So intelligent design basically urged patience, caution, and a wait and see approach, and made a very different set of predictions from evolution. In 1994, a pro-ID engineer, Forrest Mims, warned in a letter to the journal Science that they didn't publish, but he wrote the letter. He warned them not to assume that junk DNA was useless. In 1998, a well-known ID theorist, William Dembski, predicted that junk DNA would turn out to have function. In 2002, ID-friendly biologist Richard Sternberg wrote a paper on junk DNA which argued for its functions. And Sternberg's work on this topic has been praised by University of Chicago biologist James Shapiro. In 2004, pro-ID biologist Jonathan Wells wrote that people asking research questions in an ID framework would presumably have been looking for functions of non-coding regions of DNA all along, and we might now know considerably more about them, essentially, if we were using an ID paradigm. These quotes are also available in my key quotes on junk DNA post on Evolution News. So why did ID predict that the junk would turn out to be largely functional? Well, ID is a historical science which uses the scientific method to make testable predictions. As such, ID begins with observations about what intelligent agents do when they design things. And ID proponents observe that when intelligent agents act, they tend to do so for a purpose, and they make things for a purpose or a function. So it follows that ID predicts that if the genome was designed, then it was designed for a purpose. Now, ID accepts mutations as real processes, but it would say that it's not the dominant process that generated the sequence of nucleotides in our genomes. And we would anticipate that if the genome was designed, then purpose and function will be prevalent, and the preponderance of the genome would be predominantly functional. Yes, there may be some noise, but essentially the ratio of function and non-function would be very, very high. The noise would be swamped out by the signal of functionality. For me, a very eye-opening moment and this debate came in 2011 when Jonathan Wells published a book titled The Myth of Junk DNA, which cited hundreds of scientific papers, many of them published in just the late 2000s, which had uncovered function for junk DNA. But the following year, 2012, something happened that changed this debate. There was a major Nature paper published by the ENCODE Consortium, a group of hundreds of scientists, biologists working together internationally. I'm sure Dr. Dan wants to get into this. We're going to talk about this quite a bit tonight, I'm sure. And they reported evidence of biochemical function for 80% of the human genome. It basically found that the vast majority of our genome is being transcribed into RNA in some cell types in some certain circumstances. And lead ENCODE scientists predicted that with future research, that 80% would go to 100%, since they said almost every nucleotide is associated with a function. In the wake of this research, the journal Science published an article titled ENCODE Project Writes Eulogy for Junk DNA, which stated that these findings, quote, sound the death knell for the idea that our DNA is mostly littered with useless bases, unquote. And I would argue that the results of the ENCODE project are prima facie evidence of mass functionality in the genome. Again, I'm sure we'll debate this further, but I believe this is a highly defensible position. In fact, an ID-friendly biologist in an Ivy League school, I'm going to remain anonymous tonight, who studies RNA told me last week that the view in their field is basically if it's transcribed, then it has a function. So rank and file biologists who study RNA, they see transcription as prima facie evidence that it's doing something useful. Now, the decades since ENCODE's first paper saw an explosion of more papers finding specific functions for junk DNA and seriously eroded further the idea of junk DNA. In 2021, Nature reported a paper titled by Gates et al. That over 130,000 specific genomic elements, quote, previously called junk DNA, have seen specific functions identified. A 2021 paper in Genome Biology and Evolution said, quote, the days of junk DNA are over. Last year, a paper in Bioassays titled A Kuhnian Revolution in Molecular Biology, Most Genes in Complex Organisms Express Regulatory RNAs, declared we've seen a paradigm shift away from the concept of junk DNA. This was totally unforeseen by the evolutionary paradigm and totally predicted by intelligent design. And in fact, one of the authors of that paper, John Maddock, wrote a book last year by Taylor and Francis, which said, quote, the genomes of humans and other complex organisms are not full of junk, but are rather highly compact information suites that are largely devoted to specification of regulatory RNAs. These drive the trajectories of differentiation and development, underpin brain function, and convey transgenerational memory of experience, much of it contrary to long-held conceptions of genetic programming and the dogmas of evolutionary theory, unquote. So what is all this junk DNA doing? Well, we also took about a day and a half this week, me and two of my colleagues, Jonathan McClatchy and Richard Sternberg, and we went through some of our archives and spent a little bit of time and posted a link on Evolution News that's up there right now with over 800 peer-reviewed scientific papers that have reported function for junk DNA. And this is literally just scratching the surface. So what is the junk DNA doing? It's helping to form body plans. It's controlling development, controlling limb formation, controlling the cell cycle, has immune functions, metabolic functions. It's involved in brain development, DNA repair, cellular stress responses, cell differentiation. It regulates transcription and translation. And most importantly, it regulates gene expression. And sometimes the sequence of the junk is not all that important. And in those cases, DNA can serve structural functions like serving as locuses for subtelomeres, pericentromeres, and centromeres, or forming spacers around genes to protect genes. So the non-coding DNA is functioning like an operating system that is determining the timing and nature of expression of genes. So 8% of our DNA may not code for proteins, but the bulk of it is controlling the part that does. And if you want to see documentation of what it's doing, see our list of over 800 papers. Now, Dr. Dan tonight is going to argue that even if most of the genome is being transcribed, it's not producing junk DNA. It's not junk DNA, but it's producing junk RNA or maybe even junk protein. I would argue this is sort of a moving of the goalpost as what counts as function. And if you want to see evidence that evolution is hindering our progress into understanding the function of junk DNA or junk RNA, I predict this video is going to be a good example of this. So if Dr. Dan is right that the biologists still largely regard the genome as junk, and really nothing is a change in our view of the genome since the discoveries of ENCODE, then why do so many biologists frequently say things like, we used to say that this was junk, but now we know it's not? And again, see my blog post on key junk DNA quotes, documenting many quotes from biologists saying that we had a change in our view of what junk DNA is. So the bottom line is that a paradigm shift has taken place over junk DNA because there is such strong evidence of mass functionality for junk DNA in our genome. And I would argue that ID predicted this paradigm shift while evolution did not. So my apologies for going a couple minutes long, but thank you.
Thank you, Dr. Luskin. Yeah, I'm gonna for the interest of equal time, Dr. Dan, take between 15 minutes or what you need for an opening, because that did run a little bit more than 10 minutes. However, I have watched Dr. Dan's videos on ENCODE, and I'm pretty sure that he has a little bit to say about it. So let's give him the floor.
Yeah, well,
good guess, right? Good guess.
We'll get there. Don't worry, everybody. So first, thank you, Steve, for hosting this. Thank you, Casey, for agreeing to join me to chat about this. I've been looking forward to this since we agreed to it. For everybody listening, I apologize for inflicting my voice upon you. It's allergy season for me. I'm going to make a joke. My not very intelligently designed immune system thinks birch pollen is trying to kill me, so it's angry about life right now. But had to go for one. Okay, so thank you for all of that. I enjoyed hearing all of that. I took a couple notes on everything Casey was saying, and as you might expect, we're going to have plenty to talk about tonight. So The first thing I want to do is agree with Casey that, and I love when we can agree on things, is to say that while, this is a really interesting topic, it's something I love to talk about. When it comes to like evolution versus creationism, this is kind of like a little side thing that doesn't actually directly have an implication. And I bet we're going to hear tonight, kind of related to that, we're going to hear the phrase, if ENCODE is right, then evolution is wrong. And that's a quote from the aforementioned Dr. Dan Grauer. He made that at a conference talk in 2013. And fun story, Casey, I think you'll appreciate this. I was at that talk as a grad student. I was in the room for that. That's kind of a little fun little thing there. So he explains the math behind that statement in a 2017 paper, talking about how basically evolutionary processes limit the amount of the human genome that can possibly be functional. That paper was corrected in a 2020 paper that basically those authors found an error in Dan Grauer's math. Dan Grauer reviewed that paper, agreed, and if you look up his 2017 paper, there's actually a correction appended to it now. So he actually doesn't hold that position anymore, which again, this is kind of to the side of the broader thing, but just to kind of introduce the idea that when we're talking about this, I think it's relevant for predictions, but it's not directly relevant for like evolution versus creationism writ large, which is what a lot of people want to make this into. And while it's fun, it's just not that. So let's talk about junk DNA. First, let's define junk DNA. I think we can agree on just junk DNA is just DNA in our genome. It's regions of our genome that are not functional. Now, that's a really simple definition, but of course, we need to define functional then. So what is functional? Functional means that your DNA sequence, whatever it is, it's contributing to the physiology of the cell in some way that helps it survive, accomplish its, whatever it's doing in the context, like for a multicellular organism, right? It helps the cells do what they do. That's what we mean by functional. So junk DNA is DNA that doesn't affirmatively help a cell do what it does. In other words, it's a region of the genome that you could lose, and it's not going to cost the cell anything. The cell's not going to die or be hindered in some way through the loss of that region. If that's the case, we can call it junk DNA. Now, that's different from affects processes going on in the cell, affects the physiology of the cell, interacts with things, right? As we're going to see, a lot of the products of junk DNA still have effects within the cell. They're just not affirmatively contributing to that cell being able to do what it does. Now, what's required? The next thing we have to answer, if the question is how much of the genome is junk, then we have to say what's required for something to be functional, right? What criteria do you have to meet? And there's really two criteria, just like biochemically, that in order for a region of the genome to be functional, you have to check these boxes. One, you have to have the right sequence. Most sequences or most functions are going to require some degree of sequence specificity. It doesn't have to be perfect, right? There's a lot of leeway in terms of like what could be a promoter, for example, but you have to be in the ballpark in terms of being able to accomplish that specific function. There are functions like structural functions, and actually Casey mentioned a few of them, that where it's the size of the region that matters, and you have to have a certain number of nucleotides there, but the sequence doesn't matter. And we've pretty much documented what those are, and we know what those are and where the sizes are required and kind of what those are. Aside from those kind of structural functions, you need some degree of sequence specificity in order to do whatever function it is that you do. The second criteria that you have to meet is that you have to have a sufficient level of biochemical activity. Basically, you have to be doing enough at a high enough rate to matter within the cell. We tend to think of cells as like, you look at your textbook diagrams and they're like, you've got, your nucleus and your other organelles and just kind of like an empty space where you can imagine like proteins and RNA moving back and forth. But the reality is that the inside of a cell is much more like one of those, you ever see like on 100-degree day in like a city, you see a swimming pool and it's just like shoulder to shoulder, just full of bodies. That's what the inside of a cell is really like. So in order to have an effect as like a molecule of RNA, you have to be there at a high enough frequency in order to actually be able to interact with the things you interact with. Most of the genome, as I'm going to show tonight, doesn't meet those two criteria. Now, before I get into my actual arguments, I want to just take a little detour here, and I want to be clear about the arguments I'm not making. So first, I am not arguing about what a designer would or would not do, right? I'm not trying to get into some hypothetical intelligent designer's mind and say, well, a designer wouldn't make a genome that's full of junk, or a designer would make an efficient genome that doesn't have a lot of junk. I'm not making that kind of argument. I'm also not making an argument from ignorance in the form of, we have this region, we don't know what it does, therefore it's junk. If you go back 25 years, that was something we could do for a lot of the genome because we just didn't know most of what was there. But now we know what's in there. It's 2024. We've really dug into this stuff, and with the advent of really fast genome sequencing and large scale genomics, we know what's in there. So we're not making that argument for ignorance kind of argument here. I'm also not making an evolutionary argument based on genome conservation. A relatively small fraction of the genome exhibits conservation across broad phylogenetic groups. So like, for example, if you look at all mammals, only about 5% of the genome is conserved. That's different from being similar or having homology. It's a related concept, but it's different. And I'm not arguing that because only 5% of the mammal genome is conserved, the other 95% is junk. It's not what I'm doing. I'm also not arguing that we don't find function across all different types of DNA, all different classes of DNA, right? So I'm not saying that just because something is an endogenous retrovirus, for example, that means it's non-functional. You often hear something like, we've documented function in every class of DNA in the genome. And that's true. But if we're talking about, is the genome largely junk DNA, the amount of that function matters. And for most members of most classes of DNA, we actually have direct data that show that they're non-functional. They're actually junk. And I'll give some examples of that as we go. And lastly, I'm not making an argument based on what's called the C-value paradox, which is an observation that there's an enormous amount of variation in genome size between even closely related species. And this is largely due to like the amount of repetitive DNA that Casey alluded to. And kind of related to this, I'm also not invoking the onion test here, which onions have genomes that are many times larger than the human genome. So the onion test is this, it's this framing of how to think about the genome as, well, if the genome is largely functional, you need to explain why the onion needs so much more DNA than a human, right? Or even just within the genus for onions and the related species, they range from 7 billion base pairs up to 30 billion. And if there's no junk DNA, you need to be able to explain that. But I think that's a persuasive argument, but I'm not making that argument. So let me just briefly make my actual argument here, and then we can chat a bit. So first, sorry, I have to cough again. I apologize, everybody. Okay, so first, the sequences. Most functions within your genome require some degree of sequence specificity. If something is doing a critical function, you have to maintain a somewhat limited sequence in order to be able to do that thing. But we know from direct experimental data that the majority of the genome is what we call unconstrained. This means that it accumulates mutations frequently with no cost to the organism. Only about 8% of the human genome is actually constrained at the sequence level, meaning there's purifying selection operating to keep the sequences the same, right? And this is different from conserved. I mentioned earlier, this is constrained. This is referring to maintaining the same sequence in the genome. That means that the rest, 92%, can mutate freely with no cost. That means that within that 92%, they're not doing some function that requires sequence specificity. And this matters because a lot of the proposed functions for junk DNA require specific sequences, things like regulating messenger RNAs, antiviral activity, or acting as promoters. There's a limited universe of sequences that are viable to do each of these things. You have to at least be in the ballpark to accomplish them. And if you're not operating under constraint, right, if you're not subject to purifying selection, then you're not doing one of those functions. And again, I want to be clear about this. This is not an evolutionary argument. This is all about biochemical specificity. The second criterion, my second argument, has to do with the amount of activity. Most biochemical activity occurs at levels that are too low to actually be a function. So, for example, Casey mentioned that most of the genome is transcribed, and I agree. I think the kind of the best data we have right now are about 75% of the genome is transcribed. That means it goes from DNA, and then enzymes convert, they make a strand of RNA that's complementary to the template DNA strand. Now, most of that activity is in regions that don't code for proteins. The regions that code for proteins are called exons, and they make up just 1 and change percent of the genome, somewhere south of 2%. The rest of the transcription comes from things like endogenous retroviruses or transposons. These are they're sometimes called jumping genes, the little genetic elements that jump around in your genome, and pseudogenes, genes that are kind of the remnants or descendants of formerly active genes. So they're transcribed from DNA to RNA, and then supposedly the RNA does something important. But for that to be the case, there has to be enough of that RNA for there to actually be a physiological effect of it. And according to data from ENCODE that was published in 2014, a paper that was a kind of update to their very famous 2012 paper, conservatively, over half of the transcripts in the human genome are present at a copy of 1 piece of RNA per cell or less. And that's conservatively. By a more stringent measure, it's up to 90% are present at a frequency of 1 molecule of RNA per sequence per cell or less. Can an RNA that's present at a single copy have a functional role? Absolutely not. There are between 300,000 to over 1 million RNAs in the average mammalian cell at any given time. One single RNA of a sequence is not going to be able to have a functional effect on that cell. And that means we can eliminate from consideration as functional most of the transcription that occurs, and that's according to ENCODE's own data. It's simply the case that they can't have an important job to do if there aren't enough of them to do that job. So again, these assessments are based on 2 criteria for functionality: having the right sequence, having the right amount of activity. And the data shows that for the vast majority of the genome, 92%, it's not constrained at the sequence level. There's nothing keeping that sequence what it would need to be to do the function. And we also know from direct experimental data that the majority of the genome has activity at low levels. Specifically for the 75% that's transcribed, conservatively, over half of that is at 1 RNA per cell or less. This means that most human DNA not just isn't functional, but it can't be functional. It's just physically, biochemically will be unable to do the purported functions. And on those grounds, we can reliably classify most of the human genome as junk DNA.
Thank you. And I really want both of you to have a back and forth on that particular point, especially because what I've been getting for feedback has been the types of terminology used when it comes to what junk DNA actually really entails and the functionality within the cell based upon spurious transcription rates. Not random, but spurious, whether it actually has an actual effect on the cell. So people have been messaging me that seems to be like the crux of this particular debate. I don't know if both of you agree on that, but it really, to me, seems like how ENCODE determined what was functional, whether it was based on conformational changes, transcription levels, regulatory function, whatever. That seems to be the crux of what people really want to know about this particular debate. So when you guys have your back and forth, if you guys can at least touch on that a little bit, I'm quite sure the audience would love to hear about that.
Sure, absolutely. Is it okay if I respond, Dan?
Please, okay, go ahead. Yeah,
right,
I figure you'll go, then we'll just kind of bounce back and forth.
Sure. So first of all, thank you for your very cordial arguments. I had some folks worried that this is going to become a gym tour Dave Farina dynamic, and I thought
that's the debate. Wrong channel.
No, we're not like that. And Dr. Dan, I can already tell you're much nicer, and I think I'm a nice person as well. So
can I just say, I try to convince people. People don't want to talk to me. I'm nice. Steve, you watched the chat I had with Dr. Rana. I'm nice.
I literally watched that today, and I didn't know it existed because people know that I'm a big fan of Dr. Rana. We go way back. Great guy. And I got to tell you, I recommend to my audience, if you want to watch one video about this, watch the discussion with Dr. Dan and Dr. Rana. It was fantastic. I mean, I think you guys touched on pretty much everything. Yeah.
All right, so let's dive into some more detail here. I agree with you that this is not like an absolute decider of the debate over ID and evolution, but I do think that, you said it doesn't have an implication. I think there are implications that are important here, but that's really not the main issue. Let's talk about Kellis et al. 2014, this paper which you cited in response to our a video the Discovery Institute posted called "Long Story Short." It was a video we put up about a month ago on junk DNA, and you posted some responses to that and cited this paper by some ENCODE authors, one of which is Kellis. And I think the key quote in that paper is as follows. They said, for polyadenylated RNA, where it is possible to estimate abundance levels, 70% of the documented coverage is below approximately 1 transcript per cell. Is that pretty much one of the key statements that they're making, Dan? Would you agree with that? Or is there anything else that...
So that's kind of one piece of the data. They go on to talk about non-polyadenylated probably being lower, but it's harder to estimate because it's hard to isolate non-polyadenylated RNA. But then later in it, they actually talk about the criteria specifically with RNA. And I wouldn't say there's like one key line necessarily, but there's specifically a figure where they basically say in the text, they say what we would expect for spurious transcription is both low levels of transcription and low sequence conservation, right? They'd say those are the two things we would expect for spurious transcription. And then if you go to figure 3 and then figure 2 in that paper, they show that most of their transcripts meet those criteria. I think that's really the key thing.
Okay, so there's quite a bit to say in response to these arguments. First of all, we have to understand that ENCODE sequenced many different cell types. And so they were looking at lots of different cell types and lots of populations of cells. And so it's very difficult often to catch a particular genetic element in action. Okay, so this is actually one of the things that ENCODE talked about. They only actually studied 147 cell types, but there are thousands of different cell types in the human body. And many genetic elements are only active at certain stages of the cell cycle or certain stages of the life cycle. So you might have a particular quote unquote junk DNA element, non-coding element, which may only be active at a very rare time in the lifespan of an organism, maybe like, very in only certain cells at very rare stages of the organism's life cycle. And to catch it in action could be very, very difficult. So what's interesting to me is the fact that they looked at these cells, at all these different cells, and they found that this particular element is present at less than a rate of 1 copy per cell. Now, how can you have a rate of less than 1 copy per cell? It's like, when they say that everybody needs to have 2.4 children to maintain the population size. You can't have 2.4 children, right? These are averages. So they're averaging out the data that they have found looking across many different cells and cell types. And they found, oh, this particular genetic element on average is present in only, basically at a copy rate of less than 1 copy per cell. However, we all understand how averages work. It could be that particular genetic element is simply not active in, say, 145 of the 147 cell types that ENCODE is studying. And in 2 of those cell types, there are 50 copies present. Okay. That would give you an average of less than 1 copy per cell if we're looking at 147 different cell types. And in those cells where it is active, it may be very, very important. So we have to understand that the methodology here does not at all preclude the statement that it is, there's coverage of less than 1 transcript per cell in no way precludes that there are some cells and some stages of the life cycle where in fact that transcript is highly active and has a very important functional role. Okay, but we're kind of just getting started here. There's actually even more to say. And that is that there are absolutely examples of transcripts that can have activity at very low copy numbers, copy rates. Okay. And so this idea, the second criterion that's being put forth, that basically, less than 1 copy per cell on average is not a sufficiently high rate of activity. I think, Dr. Dan, I think that you said that is absolutely not possible to have function at that low rate of activity. I'm finding papers that say the exact opposite, okay, that it actually is possible. And there was a paper by Mercer et al., 2012, that found that there's a HOX-A transcript, which of course is a regulatory gene, that was found at the rate of 0.13 transcripts per cell. Now, HOX-A, of course, is a regulatory gene. and you're making transcripts of this at a very low rate. Regulatory RNAs, regulatory genes, you don't want to have them active all the time, right? I mean, otherwise you might be growing eyes out of your forehead, or you're going to trigger regulatory developmental sequences when you don't want to trigger them. Okay. So you absolutely need these transcripts. And, because a lot of these transcripts are often initiating cascades, they can be present in very, very low copy numbers to initiate the developmental gene regulatory networks, which are these very highly complex networks of genes that interact with each other to govern development of a multicellular organism. And it often starts off with one gene and maybe a very low number of transcripts, which are basically catapulting that entire network into action. But it starts off very slow and very low rates. And you may not want that transcript to be active at any other point in the life cycle other than that very specific stage of development, where you're going to then, trigger that developmental cascade to do whatever it needs to do. So now, if you're averaging lots of cells, yeah, you're going to find that HOXA is present at less than one transcript per cell on average. Absolutely, you're going to find that. But in those very specific cells where HOXA is doing its thing, I mean, it's very important for development. You've got to have that HOX gene very active. You got to have many transcripts of it, or maybe even just a few transcripts of it. Either way, we can get these low averages and yet still have very high activity. So I absolutely see no reason to accept the claim that a low average rate of presence of copy numbers has any, way to negate the possibility of function being present. In fact, there was an article in Nature Methods by Marks 2022, quoting a biologist who said that some long coding RNAs that they know are functional, we know what they do, that they have, quote, low abundance and low expression. And it said that these link RNAs, these are long non-coding RNAs. These are basically very long sequences of DNA that encode RNAs that then do not become transcripts that encode proteins, okay? And these long non-coding RNAs are basically an end unto themselves. They're performing all kinds of important functions in the cell. You can go to our blog post on Evolution News if you want to see some of the things they're doing. Very important for gene expression, gene regulation, many things like that. This paper in Nature Methods quoted a scientist saying that these long non-coding RNAs can, quote, punch above their weight, unquote, and act in non-stoichiometric ways to amplify their effects. And so this means that you are dealing with these long non-coding RNAs can have very low copy numbers and yet still have important functions. And I mentioned a book by John Maddock and Amaral, 2023. The title of the book is RNA, the Epicenter of Genetic Information. And they talk about this question of whether or not low copy numbers is basically showing transcriptional noise, or is it function? And they say that, quote, the low level and fragmentary signals from link RNAs and sequencing data sets is mainly a consequence of their highly developmental stage-specific expression, exacerbated by insufficient sequencing depth, especially in complex tissues. They go on to say that regulatory RNAs would likewise require relatively low average expression levels, i.e. more localized expression, more dynamic control. Examples, among many others, include functionally validated chromatin-associated link RNAs detected on average in less than 10 copies per cell in populations. And they talk about various examples of regulatory RNAs that have very low copy numbers. And again, Mercer et al. 2012 talked about these HOX genes that are present in a rate of, they found that some link RNAs were present at a rate of 0.0006 transcripts per cell, indicating, this is a quote, indicating expression in only a small subpopulation of the cells sampled. And they said, we calculate HOXA to be present at an average of 0.13 transcripts per cell. So the point of all this is that this idea that many parts of our genome are only expressed they're expressed in low copy numbers on average does not mean that these cannot be highly functional in certain cell types and certain stages of the life cycle. It may be very important to keep them in low copy number. And we have evidence for things we know that are functional that are present only in low copy number. So I don't see why this is a good argument that this is non-functional.
Thank you. Okay. A lot to cover there. There's a couple things I want to say briefly, and then I want to go back to a bunch of stuff where I think we agree. But first, I feel like what we're doing with the Kellis data is now I think we're extrapolating beyond what we could say, because we're looking at not a few RNAs, we're looking at the majority of transcription functional, or present at extremely low levels. Like HOXA, I mean, HOXA is a protein coding gene. So that's going to be considerably different than like a non-coding RNA derived from a retrotransposon, for example, which is a lot of, the activity that we're looking at when we're looking at transcription. There's also a big difference in functions where you're acting on a diploid genome, because a diploid genome, right, if you're regulating a specific gene that has relatively few copies, you can certainly work at relatively low levels to do that. If you're operating against mRNAs, then that's going to be quite different in terms of the number of targets. But I want to kind of agree with you about a bunch of the DNA. And actually, if I may, what I'm going to do is I'm going to change sides during this debate. I'm going to, you know what? I'm going to be on your side. I'm going to try to get the amount of function in the genome as high as possible. And before we do this, I want to put all our cards on the table here. So you've, in the past and also today, cited you and Bernie approvingly his statement that we have, we've documented 80% functionality, and he has said that he said, and that'll go to 100% at some point. You've also approvingly cited his statement, paraphrasing him, something to the effect of, we can say that there's every part of the genome regulates a different part, other parts of the genome, right? So what I want to ask you is, what kind of number do you land on in terms of how much of the human genome is functional versus how much of that is junk? And to be fair, I'm going to tell you my answer to that question, because I don't think it's fair for me to ask that question and not provide an answer. So I think we have really strong evidence for 8 to 10% of the genome being functional. And I think if you are really generous, you could stretch that to 20, and maybe you could get over 20%. Maybe you could get 25%. So I would be willing for this conversation to say that you could say 25% of the genome functional, 75% of the genome non-functional. That's where I'm at. 25 helps the cell do its thing. 75% does not. And I want to know what your number is, because what I want to do is for us to work together to get as close to your number as possible. I want to go through the genome and build up as much function as we can and get to your number. So what's your number?
Okay, well, we can certainly talk about that. I appreciate you wanting to help me out here, Dan. I'll take that as a gesture of kindness here. Yeah, I mean, I think I feel a little bit suspicious, but I'm gonna give you the benefit of the doubt here. No, but before we go into that, I do want to say, I just want to repeat that you said earlier that this expression level of below one copy number per cell means that these genetic elements, quote, absolutely not, unquote, those are your words, they absolutely not can have any function. And I believe I showed that that is not the case, that they can't have function at low expression numbers. And we see examples of this, and these are from very credible sources. So I think that argument, the main argument that you're saying shows that the genome is largely not junk, or largely, basically not helping us, largely junk. Again, this gets back to the assumption that I talked about earlier, that if we don't know what it's doing, evolutionists tend to assume that it's probably not doing anything. And this is that when we throw that assumption out the window, that's when we get papers that are actually discovering the function for this. So what exact percentage of the genome do I think is functional? Well, I honestly don't know.
Before you answer that, can I just say that's not what I'm doing? And I'm going to show that as we go through.
Okay,
sorry to interrupt.
Fair enough. You can speak for yourself. Sure, that's fine. So I honestly don't know. If I was to tell you a number, I'd be taking something out of my brain that I don't have. Like, I don't have a place in my brain to go and say, I think that this percentage is functional. Now, I do find it interesting that you and Bernie, who was one of the authors on the Kellis et al. Paper, said right around the time that paper was published that, quote, there is not a single place in the genome that doesn't have something that you might think could be controlling something else.
That's sort
of Kellis doubling down. I'm sorry, that's sort of you and Bernie doubling down on his previous quote that I think he made when ENCODE first came out, that, eventually we're going to find 100% of the genome is functional. I mean, I don't actually think that we're going to find that 100% of the genome is functional. And here's the reason why. If I take my laptop obviously was intelligently designed, although if I told you about all the problems it's been having, you might wonder. But if I were to take my laptop, put it on the top of Mount Everest for 30 years and come back, it's not going to work anymore, okay? So designed objects can have histories. And this sort of reflects the way that intelligent design theorists approach science. We try to look at the way, what do we observe in the natural world around us in designed objects? How does history affect their behavior? How do intelligent agents design things? And we see that very frequently designed objects have histories. They might be aboriginally designed, and yet then over time, natural processes destroy the function of those designs. And I do actually think that in some cases, pseudogenes could be examples of that. You have examples of genes that one time did have a function, and they no longer do. They accumulated mutations, natural processes destroyed those functions. I think that's possible. Now, there's a lot of other things to say because a lot of pseudogenes are turning out to have functions. So I think we have to be very careful about going there, but I have no problem with that. So I'm definitely below 100%, no question there. I think though that we're talking about ratios here. Is the ratio of junk to function, basically very, very high, or is it very low? I think that intelligent design is going to predict that it's going to be on the lower end. Okay. Obviously you come down on the higher end. You think that maybe only, and I don't want to misquote you, but it was like 8 to 10% probably is what we're looking at. So, you're looking at a ratio of maybe 9 to 1 of junk to non-junk, somewhere in that range. And, I don't know exactly what the final number is going to be. What I do know is this, is that as an intelligent design theorist, I am incredibly encouraged by the data that has come out over the last 10 to 15 years. Because remember, I told that story of the Idea Club, where back then, nobody could cite papers of function for junk DNA because we hadn't done hardly any research yet. And now, we put together a blog post of 800 papers that have found functions for specific non-coding junk elements in just a day and a half, and I was actually doing a million other things prepping for this debate during that time. So, it was not hard to do that. Now it's just a tiny snippet. It's a tip of the iceberg. And, I've seen people citing, there's a paper, Gates et al. In Nature that shows this curve of papers that have discovered function for junk DNA. And that curve is like basically there's two curves in this diagram. A curve showing our knowledge of the number of genes, and it sort of peaks out around 2003 when the Human Genome Project happened, and then papers showing our knowledge of the number, or I guess our knowledge of the number of specific genetic elements. And that curve is going up at an exponential rate. So our knowledge of function for junk DNA is just going up, 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 up. And this is exactly what ID predicted. Now, there's a lot we don't know, and I'm going to stop talking soon. But I don't know if I can give you what you're asking for, Dan, because I honestly don't know. I will say this, is that the trend line of the data, and the data shows, and this trend line shows no sign of slowing down, is exactly what ID would predict. And I'm very excited about that. If you want to see the Gates et al. Figure, watch our long story short video that Steve has linked from the description of this video, which we got permission to actually show that figure in the video. And it's a nice figure. It shows the rate at which our knowledge of function for junk DNA is going up, and it looks like an exponential curve, and it shows no sign of slowing down.
All right. Well, so let me, rather than try to, pry for a specific number, then let me just, how about we say conservatively that 50-50, half junk, half function, I think would be a conservative estimate for most ID proponents. That sound fair to you?
I would say it's probably going to be greater than 50%. I don't
Greater
know
than 50%.
But if that helps you out, I'm expecting it to be greater than 50%.
Okay,
I'll go with that. Yeah, I'll go with
let's
that.
Say, all right. So what like I said, I like to start with stuff that we agree. I like to do stuff we agree on. So let's take that number as 50% as kind of like our baseline, like that would be a high amount of functionality. And I think in reality, most ID proponents think it's going to land somewhere north of 50%, but we can do 50%. So I do want to say, I think that trend line is slowing down. I'm going to provide some data to that effect in just a minute, but let's start with the stuff we agree with first, because I bet we'll disagree about that. So I'm going to go through just real quick I know you know a lot of this stuff. I'm going to go through for the audience, kind of what the genome is made of that we know the function for, stuff that we know is functional, right? And let's see how high we can get that number that we agree on. So we have things like, and I'm going to start with the really small things, and I'll get bigger and bigger as we go. So regulatory regions that are really short in the genome, things like promoters, where you start your process of transcription that's going from DNA to RNA, things like enhancers and silencers that affect the level of transcription. Collectively, that's a fraction of a percent, but I'm going to be really generous and just round that up to 1%. I'm going to just round up to the nearest percent when I have the option. Similar story with origins of replication, so places where DNA replication starts. It's a small fraction of a percent, but we'll just say 1% for those. Then you have your exons, your protein coding regions of your genome. That's like 1.5%, plus you have the short untranslated regions on either end of those that end up in the molecule called messenger RNA that uses a template to make proteins. With the UTRs, the untranslated regions, we can be really generous with that, and we'll call it 3%. It's closer to 2, and we'll call it 3. Then you have your RNA genes. So these are going to be genes that are expressed as RNA, but they don't get translated to protein. So this is going to be like your transfer RNAs, your ribosomal RNAs, your long non-coding RNAs. We can round up to 3% for those. And all of those regions that I just described, those are highly constrained regions of the genome in terms of having, sequence specificity. Then we can look at structural regions that are a little more loosey-goosey with the amount of, or the specificity of the sequence. So these are going to be things like centromeres, which are in the middle of chromosomes, telomeres that are on the ends of chromosomes. You have your scaffolding regions for like protein attachment. You have spacer regions where the sequence of the DNA doesn't matter, but it has to be a certain length, right? It can be 2,000 nucleotides, 1,000 is too few, 5,000 is too many, but as long as it's the right length, the sequence doesn't matter. You add up all that stuff in the genome, it's very well documented, that gets you about 3%. Now that puts us right there. 1, 2, 3, 4, 5, 6, 7, 8. That puts us at 11% functionality with being generous with our rounding. That gets us to 11%. Now we can go one step further than that because ENCODE documented that 8% of the genome is involved in binding to transcription factors. Transcription factors are proteins that grab onto the DNA and regulate the process of transcription, making RNA from that region of DNA. Now, within that 8%, we have kind of a lot of problems. There's a lot of redundancy with the other regions we've already described. The levels of binding have been shown to actually be very brief in some cases, so it grabs on and then it lets go. And so that's almost certainly spurious binding. And then the way that ENCODE specifically did this assay was overly sensitive. Basically, it was kind of an indirect method where they removed the enzymes that would have blocked access to those sites on the DNA and then treated the DNA with enzymes that targeted those sites. But there's other reasons those sites could be exposed. So that 8% is an overestimate for a lot of reasons, but we're not going to worry about that. We're just going to count the whole 8% and add it to our already existing 11%, which gets us to 19% functionality, which I think we can agree on that. Casey, I think we can agree on all of those things. We could say that's functional, we're good. Can we agree on that?
Well, I do question some of your numbers. So, I agree it's at least 90% functional. But, for example, I've heard statistics. Do you want me to? I don't want to cut you off.
We'll get to more. We're getting into more regions, but just like as a first pass, I think we agree.
Even some of your numbers for enhancers, for example. There have been estimates that there are up to 400,000 enhancers in the human genome, and I just Googled it. Enhancers can be anywhere between 100 to 1,000 base pairs in length, and so let's just split it down the middle and call it, 550. Okay, that's over 200 million base pairs, which is about, it's about 7% of the genome right there that could be enhancers. If you go with, out of our 3 billion base pairs of the haplogenome. So, I think that, or half, the coding strands. So I would say that I don't know if I fully assent to all of your numbers there. Some of them I probably do.
Right,
but some of them I think that we are dealing with cases where this is what we know at the present time. And there's a lot more yet to be discovered. And we're still discovering what these genetic elements are doing.
I'm good with it. Fine. Let's make it, like I said, I'm here to help you. Let's get this number as high as possible. We're making that 7%. All right. So
but it's not just enhancers. I think that there's a lot of examples of.
Oh, we'll get there. We'll get there.
We don't know. Anyway,
let's keep going, because the rest of the genome, we know what it's made of. And the question is it functional, right? So we've got our retrotransposons, are about 45% of the genome. These are RNA-based kind of copy and paste little things that just move around the genome. You get your DNA transposons. Those are cut and paste, similar, but they're cut and paste rather than copy and paste. They're only 3%. Then you've got your endogenous retroviruses, 8%, pseudogenes, 5%. You put all that stuff together, that's 60% of the genome right there. And that's where a lot of the transcriptional activity is. And that's where a lot of the purported function is from people who say that there is no or very little junk DNA. A lot of it has to come from that 60%. So we can just like, we have data on this. And this is why I disagree with the argument from ignorance thing here. Like, we have very specific data on this. And I'll just use one example of this. There are these elements in the genome called lines, L-I-N-E. It's an acronym. It's all capitalized. Lines stands for long interspersed elements. They collectively make up about 21% of the genome. It's a class of retrotransposon. Now, of that 21%, there's actually 17% of it is just a ton of copies of this one specific thing called L1 lines. And we've documented function in L1 lines. We've actually documented specific things that they do, because they actually, when they're intact, when they have all of their parts, they actually do a lot of things. They do transcription. They do a bunch of things. They do reverse transcription. They can cut DNA. They can do a lot of stuff when they have all their parts. So we've documented function in about 100 of them, which is really impressive, except that there are about 500,000 L1 lines in the human genome. And it's not that we haven't looked at the rest of them. We have, and they're not doing the functions that the 100 functional ones do. And in many cases, it's not just that they're not doing those functions, it's that they can't do those functions because they're mutated and they've experienced deletions because they're not sequence constrained. So they've actually broken down to the point where they can't do them. So we're not out of ignorance saying that we have 100 functional and the rest aren't. It's out of knowledge, out of direct experimental data, that we're saying we found 100 of these that are functional. And then we have approximately 499,900 of them that are not. And so we can add, like, taking those 100, they're not big things, but we can take those and round up to 1% and add another 1% to our tally. But at the same time, then we have to add 16% to the side of the ledger that's junk DNA, because, again, I want to be clear about this. It's not just that we don't think they're doing the function. It's that for the most part, those sequences are so degraded, they can't do the function, the regulatory things, the affect the structure of DNA things that the functional ones are doing, right? So that's going to be a theme here as we go through these.
So let's talk about this idea that some genetic elements are too degraded to be functional. I don't know what a degraded line is supposed to be, but I do know what you talked about, because, what is an aboriginal line supposed to look like? I do know that you've talked about degraded endogenous retroviruses, and the idea that, some ERVs might be able to produce some kind of, mRNA transcript that can have some kind of a function, but many of them have these long terminal repeats. That's all that's there, and basically the coding sequence of the ERV has been degraded. Well, there absolutely are examples of ERVs that only have where the LTRs are functional, and the rest of the ERV is degraded. In fact, there was a study that found that ERVs these kinds of LTR ERVs can function as enhancers. Excuse me. And ERVs in general are known to be highly functional. There were 2 studies on mouse embryogenesis from last year that found that hundreds of mouse ERVs are necessary for development. So again, I don't know how you are making this assertion that, all these line elements are too degraded to have function. I don't know what your basis for that is. To me, this sounds like, once again, we are assuming that it doesn't look functional, therefore it must not be. Evolution assumes that random mutation is building our genomes, and these look like they are degraded, quote unquote, but therefore they must be the result of just mutations screwing things up. I would argue that these ERVs that are basically just, they're not actually degraded LTRs. Those LTRs are there for a function and a purpose. And I just don't see that. These are very different visions of the genome that you are presenting versus what I am presenting. And we can sometimes find what I would call circumstantial evidence of function. There was a paper that I believe was in Nature reporting the mouse genome that found hugely convergent insertion, points for signs, which, as you said, what percentage of the genome, remind me, are sign elements?
It's about 21% are sign elements, and yeah, they are. And because just to for the audience, because this is very specific, it's these genomic elements called retrotransposons. There's these little RNA bits that convert themselves to DNA using an enzyme and then basically cut your DNA and put themselves in there. And then they can transcribe back to RNA, and that piece of RNA can go and do it again. So yeah, these, when they're active, they are associated with things like insertions and DNA breaks and things. Yeah. Mm-hmm.
So these signs are thought to be inserted into the genome.
Oh,
As
sorry.
you said.
Just to be clear, those you just mentioned signs, that's a different thing.
I know, but I'm saying
Okay.
that's 20% of the genome right here.
Mm-hmm.
And we see a signal between rat and mouse that the insertion points for signs are convergently very similar. And to me, that's very interesting. That shows, I think, evidence that there's some reason for why they are where they are, and they probably have a function. So again, I just don't know on what basis are you asserting that line elements are functional. And I would also ask you this
I can
question,
tell you.
since you asked me a question. At what point would there be enough peer-reviewed papers? There's tens of thousands of peer-reviewed papers that have found function for non-coding DNA. At what point would there be enough papers for you to say, okay, look, the trend line of the data shows that my default assumption that this is probably junk, I shouldn't use that default assumption. Even if I think that there's some evidence there for it, this assumption ought to be very dubious. At what point would you be willing to question that assumption?
Okay, so for that last bit, I'll answer that first, and then I'll go to all the rest. It's not about like a number of papers. It's about the weight of the data, right? And like what part of the genome is specifically shown to have a function experimentally versus what part is shown to not have a function, which is different from not yet being shown to have a function, right? And what I'm describing with these, bringing it back to the line elements, is we know they don't have a function because the ones that are functional, they have very specific pieces of them. And this is also true for endogenous retroviruses, which are the remnants of viruses that insert themselves into the genome and then kind of get stuck. The different parts of them do very specific things, right? So there's an endonuclease, right, which is an enzyme that cuts your DNA. There's a reverse transcriptase, which is what converts it from RNA back to DNA, right? There's the promoter region that allows it to get out, right, to transcribe it. Now, for the lines that are non-functional, the 499,900 of them, the vast majority of those, when I say degraded, I mean they've either experienced mutations that disrupt the structure of those regions, that disrupt the sequence of those regions, so they're not capable of, for example, acting as a promoter to initiate transcription. Or if you were to transcribe and translate the line gene from one of their enzymes, the mutations would make it so that that enzyme no longer does the specific activity that it does in the kind of intact versions, right? That's what I mean when I say degraded. It's that it's been mutated or it's experienced deletions to where the functional parts of it physically cannot do the job that they do in the functional examples. And the same is true for endogenous retroviruses. So the structure of an endogenous retrovirus, this is a virus, everybody, that inserts itself into your DNA and then gets into your germline cells, your reproductive cells. So this little bit of virus in your genome gets passed on to your offspring. And now it's called an endogenous retrovirus. An example of a virus that could potentially do this is HIV. HIV inserts itself into DNA. When someone gets HIV, it goes into your DNA. So these endogenous retroviruses, the structure of them is they have two regions called long terminal repeats. And then in between, they have three genes. And we can name the genes. It doesn't matter what the genes are named. It doesn't matter. There have been a few documented examples. And when I say a few, I mean single to maybe double digits, where we've actually, our genome, the human genome, has co-opted one or more of those genes. The most famous example works during human development and is the reason that we can have placentas that last throughout development instead of the embryo getting evicted early in development like what happens in marsupials. That's the most well-known example of this. There's a few other examples of endogenous retrovirus genes being kind of acquired as part of the human genome. There are more examples, many more examples, where those LTRs are what matters because those LTRs are the promoters for the genes. And when a retrovirus lands in your genome near a gene, its promoter then causes transcription of that gene or the adjacent region, even if it's not a gene. But in order to do these functions, the sequence has to be close enough. It has to be recognizable as a promoter. the genes that we're going to co-opt have to be present. Most endogenous retroviruses are what we call solo LTRs. They've lost that whole structure except for one of the LTRs. Sometimes those LTRs still function as promoters, and we've like kind of co-opted them. And those are absolutely examples of functional endogenous retroviruses.
Okay,
but they just like one or two sentences. They make up a minuscule fraction of the 200,000 endogenous retroviral sequences in the human genome. So of that 8%, to assign functionality to 1% of it would be extremely generous. And again, I just want to emphasize before turning it back over, this is not out of ignorance that we're saying the other 7% is non-functional. It's because we know what those sequences are, and we can see that they're missing the parts that are required to be functional. So that's another example of where we can assign a little bit more to that functional fraction, but proportionally a lot more to the junk fraction.
I think what we're seeing on display here, Dan, is the same kind of thinking that led people to presume that the non-coding DNA was junk for decades. You don't know what it's doing. You don't know. You don't actually know that it's not functional, okay? You know that it's not doing its normal, job that it should be as far as being able to provide transcripts that can serve to insert it into other places. But as I explained with those examples with the ERVs, these LTRs absolutely can be quote unquote degraded from what we think their function is supposed to be, but they can still have functions as enhancers. And there's absolutely the possibility that these degraded line sequences could have functions that we are not expecting. They could be attracting all kinds of different things. I mean, line elements have lots of different functions. They're involved in gene silencing. They're involved in chromosome inactivation. They're involved in repairing DNA breaks. And I don't know how many of those require them to have a lack of degradation, but I do know that there's lots of functions for non-coding DNA that do not require something to perform its normal quote unquote function, providing a transcript that can actually be processed or whatever. You can have attachment points for promoters, enhancers, et cetera, et cetera. And I just don't see why, and these can take all kinds of different sequences. I don't see why this has to be the case. It sounds to me like you are still making an argument from ignorance, okay. And I just don't see how you've established that there's actually evidence of a lack of function.
Okay, allow me to try again. The reason is because we have a bunch of purported functions for these things, right? So like ERVs are a really good example where one of the the main proposed function that you hear for endogenous retroviruses is that they perform an antiviral function, right? Is that you have this virus-like sequence in your genome, and then it gets expressed as RNA, and then when the viral RNA is present, the endogenous virus sequence interferes with it, and it performs an antiviral function in your cells. In order to do that, it needs to have a certain level of sequence similarity to those target sequences. So we can actually biochemically calculate the percent of sequence complementarity between those two things to mathematically answer the question, will this piece of RNA from the virus hybridize, form a double helix with this piece of RNA from your genome? And we can show, based on the biophysics of it, that is the answer to that question for most endogenous retroviral sequences is no, because either the sequence is not transcribed because the LTRs are broken down, or the transcribed sequence is too short to matter for the length of that complete virus, or the sequence has diverged from the virus by too much and it would no longer effectively hybridize with it within the cell. So again, I'm not making an argument from ignorance here. I'm there's very specific reasons why we can make these evaluations.
So maybe it's not going to
hang on one quick thing with the lines. You described, for instance, DNA break repair. Yes, that is one thing that they do. But in order to do that, they need to have the line enzymes, because one thing that they do, remember, is they cut your DNA and they stick themselves in there. So they're good at taking one piece of DNA and sticking it into another piece. And that's, in effect, double-stranded break repair. But in order to do that, they need to have the enzymes, one, present, and two, the sequence needs to be there so the enzyme will work. And for the vast majority of line sequences, that's not the case. The enzymes are either missing entirely or they're full of mutations. And I do want to point out that I think it's curious that you're a well-known intelligent design proponent, that you are in effect arguing here that you don't need a specific sequence to do these jobs. You can have tons of sequences that do these jobs.
Hold on.
That's what it sounds. That's what it sounds like.
Look, you've made a fair point that if the sequence is degraded, you can't maybe do DNA repair, or you won't be able to bind to a viral, an ERV transcript cannot bind to viral RNA or DNA or whatever, or and then be able to lock it up and prevent it from causing an infection. Okay, I that's a fair point you're making. My point, but I'm not saying that they can still perform those functions. I'm saying that there are many different functions that non-coding DNA is known to perform that can happen in these quote unquote degraded sequences. So I just don't see evidence. There's a whole plethora of other functions that could be taking place from these sequences that do not require them to have the typical, what you would call, their standard sequence. I just don't see, again, it seems like you're making an argument from ignorance again, that you don't see how it could do a function. Therefore, you're going to conclude that it isn't. But we do know examples. I mentioned earlier, the ERVs that are degraded and yet still have functions. That could be the case with many other types of genetic elements. And so I just don't find this to be an argument that's really different than what we've already seen. I don't see this as conclusive evidence for lack of function. I think that we're making assumptions still. And I'm content. Look, Dan, I'm happy to say you made an argument here. And I'm happy to say what I said 20 years ago. I'm willing to take a wait and see approach, because you know what? The evidence for function for junk DNA is piling up. And I have not heard powerful arguments that these elements don't have functions. Rather, I see thousands of papers in the literature that is finding functions for lines, for signs, for ERVs, for various forms of repetitive DNA, all of that. And it's finding it in various types, various forms. So I don't feel like it would be inappropriate for me to concede this point, because that could risk stopping science, and that could risk actually hindering research into the function for these quote unquote, degraded genetic elements. So that's what I would say. Yeah.
So I agree again, and I said this at the top. I agree that we find functions in all of these different types of DNA, whether it's retrotransposons or ERVs or pseudogenes. We do, in fact, find functions in all of them. The problem is that it's a very small fraction in which we find those functions. And when I'm talking about like it could do the function versus it can't do the function, I'm talking about the functions that are either shown to exist in these elements or are purported to exist by people who oppose the concept of junk DNA, right? So it's again, like we can take a wait and see approach, but we've got a lot of data now. And I think a wait and see approach was appropriate 25 years ago. And right now we have more data than we had 25 years ago, and 5 years, 10 years, 20 years, we're going to have even more data than we have now. I'm not saying not to look into this stuff. I'm saying for the stuff that we have well characterized and looked into, we can make very confident judgment calls about its ability to do these purported functions. But you've been saying it could do this, it could have this other thing. So let me ask you, what specific functions? Wait, hang on. No, let me just finish the question. What specific functions could these sequences be doing that do not require sequence specificity?
I'm going to get to that in a minute. I want to talk about this idea that you just said, and excuse me, I'm losing my voice here, and I'm almost down to the bottom of my Starbucks cup.
We're coming a little bit up to the 90-minute mark, but you got plenty of time. So
I'm not trying to cut it off. I'm just apologizing. So you talked about that we've studied these things enough to conclude that they function. Well, that's what I heard you just say.
I wouldn't say enough, because I don't want us to stop.
This is exactly what people said about the non-coding junk DNA 20, 30 years ago, that we know it's not doing anything. And I just feel like these are the same arguments that if I were to accede to these, I would risk becoming a science stopper. So I don't want to accede to these. You may tell me that you're not saying we should investigate this, but that is the effect of this. And let's talk about pseudogenes for a moment, okay? Many people would say that the vast majority of pseudogenes are an example of functionless junk DNA. But what I'm reading in the literature is that we've just barely begun to scratch the surface of studying pseudogenes. There was an article in Nature Reviews Genetics a few years ago by Cheatham et al. That talked about how we were just developing the technologies to study pseudogenes. And they had this really interesting quote. They said that pseudogenes that have been, this quote, pseudogenes that have been suitably investigated often exhibit functional roles. Or they say, quote, where pseudogenes have been studied directly, they are often found to have quantifiable biological roles. So what we're saying here is that when you actually take the time to go and study these genetic elements, things that everybody thinks are functional. I know many evolutionary scientists who have used pseudogenes to argue against intelligent design. I mean, I could tell a little story about this. Back during the Dover trial, the Kitzmiller versus Dover trial, Ken Miller cited a beta globin pseudogene and claimed that this pseudogene is found in, with the same molecular errors, is found in gorilla, is found in chimp, is found in human. And this is evidence that it is non-functional and that it is basically evidence of junk DNA and evidence of our common ancestry. Well, lo and behold, in 2013, there was a paper that came out that found that this beta globin pseudogene has a higher degree of conserved sequence than you would expect if it was just, accruing mutations at a neutral rate, et cetera. And so that was sort of initial evidence and function. And then I believe it was in 2021, there was an article in Cell Reports that found that this pseudogene is indispensable for creating red blood cells. Okay, now Eugenie Scott went up. She is a very famous ID critic. She went before speaking at the Natural History Museum at a symposium they had in 2007, basically attacking intelligent design, even attacking me in that lecture, saying that this pseudogene, quote, doesn't do diddly. Okay, well, somebody then took the time to go and studied it and found that it was indispensable for creating red blood cells. There are many, many anecdotes that we should be taking very seriously. And this paper in Nature Reviews Genetics said the same thing, that when we actually take the time to study pseudogenes, we are finding evidence of function. I've also found many papers in the literature which have said, again, that the technology is just at the very beginning for us to understand what these things are doing, and that really technical limitations are still at work here in preventing us from studying many of these non-coding elements. So basically, I think that what you are arguing is that, I'm hearing you say this, is that these genetic elements, clearly based on what we know, cannot be functional. And I'm saying I've heard that before and it turned out to be wrong. And I don't think we should make that mistake again. You asked me for a particular function of these degraded repetitive elements. Well, they bind transcription factors and they're associated with the regulation of neighboring genes. Their protein coding regions are not intact, but both of them have a number of cis regulatory functions. So there are functions that we can have related to regulating genes that don't require you to make a fully functional transcript, and this is known. So we have not ruled out the possibility that all this repetitive DNA is non-functional. What we do have is a huge genome, okay? 3-point-something billion base pairs for the haploid genome, right? And so what that means is that we got a lot of work to do, right? There are a lot of genetic elements that we have not, quote, suitably studied yet, as Cheatham et al. Put it in Nature Reviews Genetics. And so if we're going to start making this argument that we know conclusively that it's junk, I think we're making the same old science-stopping mistake. As I said in my opening statement, I was predicting that people were going to hear, that the science-stopping effects of the evolutionary view of junk DNA are still in full force. And I feel like I'm hearing that from you, Dan. Now, I think that you're an open-minded guy. I think that if you saw a paper discovering function, you would quickly accept that. But I think that your default assumption here is misguided, and it is taking science potentially in a very dangerous direction, which would repeat the mistakes of the past, where we are concluding that something is not functional when we really don't know that's the case.
So I think, again, that's not what I'm doing, and I can use pseudogenes as an example, and then I'll go back to the question I asked, because it's a very specific question.
I answered the question, what they do.
No, not actually. So I'll get there. So with pseudogenes, this is a great example of where we have pseudogenes, everybody, they make up 5% of the genome. And it is true that we have documented some functions in pseudogenes, like virtually all classes of DNA. We've documented some that have an important role in the cell and are under constraint to accomplish that role, whatever it is. Now, there are a bunch of jobs they could potentially do. So for example, they could be transcribed into RNA, and then they can direct that one piece of RNA can directly act on another piece of RNA to regulate its translation to a protein. You also have kind of the opposite thing where the pseudogene RNAs act as alternative targets, like decoy targets, for regulators of the mRNA, right? Where instead of grabbing onto the real mRNA, the proteins or whatever grab onto the pseudogene RNA, and that then facilitates transcription of the actual or translation of the actual RNA. And then as you alluded to, they could also work as regulatory elements actually in place in the genome, because many pseudogenes, they have, they're associated with promoters right there. And so you can have different regulatory elements associated with those pseudogenes that regulate other nearby actual protein coding genes. The thing with all of those functions that we've documented in pseudogenes is that they require sequence specificity. You need to have the two RNAs need to be similar enough to interact with each other. The one mRNA needs to be similar enough to the other one so that the proteins that would grab onto one actually see the other one as a viable target. And within the genome, to act as a promoter or an enhancer or a silencer, you need, like, those are particular sequences that would be maintained if they were actually doing that job. And most pseudogenes, we can directly document this, are not under constraint. They're not in that 8% of the genome that is constrained at the nucleotide level. Now, Casey, you just described some of the functions, right? The cis-regulating elements, and and it's true that pseudogenes do these things. But my question specifically was, what functions are proposed for these elements that don't require sequence specificity? And all the ones I just described, including the ones you described, do require some degree of sequence specificity.
Well, they may require sequence specificity, as you said, for some RNA, and that RNA goes out and regulates the gene. And you have microRNAs that are so short that it's hard to say that this has sequence specificity to some mRNA transcript. So I think that
that's a different thing, though. That's
not
pseudogenes.
What I'm saying, no, we were talking about repetitive DNA earlier, and that's what I was actually answering you about, the function of degraded repetitive DNA. You could have these short repetitive elements that could be producing RNAs that have all kinds of regulatory functions. There's no problem. You don't have to have direct sequence specificity to an RNA transcript that is an mRNA transcript that is directly encoding the protein to have a regulatory function. Okay, I'm pretty sure about that. Now, as far as some pseudogenes having a degree of sequence specificity that is so degraded that it can supposedly no longer regulate its parent gene, well, the reality is the degree of sequence specificity is going to determine how tightly it binds. It binds to the mRNA transcript. And so something with less sequence specificity may just have a lesser degree of binding ability, and that may be part of its regulatory role. So, I mean, the exact degree, the fact that it differs from the aboriginal coding transcript is not a problem at all. The fact that it may even be very different from it. Maybe it's only intended to, bind a part of it and lock that part up. There are so many possibilities here that I just don't feel like, again, we're establishing clear evidence that this is not functional.
So that last thing you said, I think, is really important, because you said there's so many possibilities here, but I think that's getting out over your skis a little bit, because now you're proposing functions that we have not directly documented. And again, the things that you're describing, like small interfering RNAs, that this is part of a mechanism where you have enzymes that grab onto little bits of RNA, and then they find that same sequence in the bigger RNA, and the enzymes cut at that sequence. So again, you need sequence specificity. But you're muted.
So I didn't have that specific function in line. I'm just saying that you don't have to have sequence specificity to an mRNA transcript in order to have a function.
Okay.
So I just don't feel like that's a requirement of being functional. That's all I'm saying.
So I think then a related thing here, because for the functions that I've been describing, you do need sequence, like not 100% sequence specificity, but you need a documentable amount of specificity in order for these different regulatory functions. I should note you also need transcription for many of these functions for pseudogenes, and many of them are just not transcribed at all. So they can only operate as regulatory elements, but not on mRNA. But that's kind of, I want to just move on. Well, go ahead, and then I'll.
Yeah, that's actually what I thought you originally were going to say. And there actually is a paper that I have cited for a long time that says that the DNA of a pseudogene can itself have function. So let me find that paper here. It is Polisano 2012 in Science Signaling. So it does not have to necessarily provide a transcript to have a function. Now, all that being said, of course, I like the idea that pseudogenes, and I should say this also, it is possible, as I said this earlier, that some pseudogenes might represent legitimate examples of junk DNA, where they are a copy of a gene that got degraded. They originally were function, or maybe there was a duplicate copy that was created through a random mutation, and you're looking at something that once had function and now does not. I don't rule that out. I also don't rule out the possibility that there are some, endogenous retroviral insertions in our genome that represent ancient viral insertions. And I do also believe that, we can observe that transposons can hop around. So, but I think that these things are probably going to turn out to have a lot more function than you're expecting. And especially in the case of transposons, where it looks like they're sort of like designed elements that help us to, this is Jim Shapiro's natural genetic engineering idea, that transposons are very important for helping to re-engineer the genome in response to environmental stress that an organism is experiencing, and can help to do a lot of basically helping an organism to overcome periods of stress. That might be, these retrotransposons these transposons may have functions in that department.
So sticking with this, whether it's pseudogene, I think pseudogene is a good example to illustrate the point I want to make here, because you said something important a few minutes ago about how, well, it doesn't have to have exactly the same sequence, and if, as the sequence is maybe a little bit different, it interacts at a slightly lower level, but it's still regulating it. And this is where I think we get into a lot of the spurious activity in the genome. That's when I say spurious, everybody, when we say that we have a sequence that has activity that is just kind of happenstance due to other factors going on, like you're expressing a sequence nearby, and then the polymerases grab onto this one. That would be an example of spurious transcription. You could have spurious interactions between different RNA molecules, but I think what we're mostly documenting with that kind of thing is something I mentioned at the very top, way back in my opening, which is where you have these molecules that are affecting processes, but they're not regulating those processes. And we can be confident that that's the case, because if we are regulating the amount of translation, right, you have an mRNA that you make from your genome, and that's going to be translated to a protein at a certain rate. If that rate is tightly constrained, then and it should be, then the things that are going to act on that mRNA that affect that rate are also going to be under tight constraint. And I don't mean sequence constraint strictly. I mean like regulatory constraint. Your cells are going to clamp down on this stuff to make sure it works the way it should. Now, those pseudogene RNAs, again, largely almost all of them are not constrained at the sequence level. So they can experience mutations. They can differ from we'll call them the target mRNAs. This is akin to, like, in my house, like I live in a really old house. It's 110 years old. My walls are not insulated. So I'm sitting here in my basement. My basement in the winter is the warmest place in the house because it's insulated. For the rest of the house, if I want the house to be 72 degrees during the winter, then I have to set my thermostat for like 75 degrees. Now, are we going to say that my drafty, uninsulated walls are regulating the temperature of my house? No, absolutely not. They're not regulating the temperature of my house. They're certainly affecting the temperature of my house, but are they functional in maintaining that temperature? No, absolutely not. And in the same way, a lot of these transcripts, both from pseudogenes and other things, are certainly affecting lots of processes that go on in the cell. But because they're not under selective constraint to maintain that sequence that affects that thing, that shows that they're not being selected to do that job, right? The sequence can wander around, which means there's no cost or benefit to what it's doing. It's just doing it kind of willy-nilly within the cell. Willy-nilly is not a good word, because there is, specificity.
Well, I mean, they're experiencing random mutations, but I don't think that you really know that that's what's happening. You're inferring that that has experienced basically neutral drift because there's no specific function, or maybe a very weakly important function, and that therefore it can accrue a lot of, basically very low selection pressure on this. Okay. You're assuming that is an assumption. And the idea also that, sequence always has to matter hugely for these non-coding elements to be functional, I also don't think that is true. There was a paper that came out not that long ago in the journal, I'm trying to look this up here, the journal Science. Short tandem repeats bind transcription factors to tune eukaryotic gene expression. Hope you guys can still understand me here. And basically, these short tandem repeats, which make up about 5% of the genome, are described as rheostats. Rheostats are kind of like your thermostat. You can fine tune them very precisely. And these short tandem repeats, you can basically, by changing the way that they are binding to transcription factors, and presumably, changes in sequence can actually cause fine tuning of various functions. And this is an example from an ID. Let me give you an ID perspective on how the genome works. We are allowed to have mutations in the genome under intelligent design. And a lot of these mutations, these ideas have come out of one of our research projects called the Engineering Research Group. A lot of mutations that take place in a design view of the genome are sort of pre-planned variation that the genome is allowed to experience to allow it to sort of modulate various switches within pre-designed parameters, okay? And so sometimes they call it environmental tracking, where you can basically have environmental cues that are, causing perhaps mutations to occur, and then the genome can respond in ways that it's pre-designed to do, and this can cause sort of switches to turn, or chase a rheostat, something on an analog continuum. You can basically change various functions in response to predetermined pathways. And so with these STRs that can basically bind transcription factors, and these are very, very short sequences, we're talking about just a few base pairs. The degree to which they will bind to a transcription factor affects their function. And this is very, very common in gene regulation. The degree to which something will bind a transcription factor or a promoter or whatever affects how the gene is being regulated. So you can allow for lots of variation in these non-coding genetic elements, which will allow you to then modulate gene expression along predetermined paths. And so I don't think that the fact that we're seeing variation or the ability of sequence to vary, that does not bother us intelligent design proponents at all. In fact, this shows us that the genome is designed to be able to experience small scale changes to modulate things within predetermined limits. And it's a very interesting aspect that we're actively looking at in genomics right now. I'm sorry, I can't talk anymore.
Yeah, so
your voice is going out a little bit.
Yeah, so and I feel like this is where we're really kind of working through this. So this might be a good time, Steve. If Casey, I don't know if you prepared like a short closing. I just have like a short summary. I've been taking notes, but I do want to respond to that just very briefly. And then if you want to maybe respond to what I said, and then if we want to do closings and call it good, because
I got a few questions to first.
Yeah,
not that many, just a few of my own and a few from the live chat.
Okay, so let me just say to kind of that last thing, we're kind of going back and forth a bunch here. I feel like between we have a ton of research that's showing that more of the genome is functional. It's more and more every year, and we're documenting more and more of these functions. But then every time I respond to it with, okay, we've shown this much is functional, I agree, but then we're also documenting this much that can't do any of these functions. It's then a lot of, it could be this, maybe it's that, maybe there's stuff we haven't found yet. And I want to be clear, I agree. I am sure that there is stuff we have not found yet. And that's why that in my opening, I said, or not in my opening, but earlier I said, I think we have really, really rock solid evidence for 8 to 10% functionality. And I bet you can get that, up to 15, 20, maybe even higher than 20%. But the key thing I want to emphasize for everybody right here is that along the way of documenting those functions that we keep finding, we also look into lots of the rest of the genome that can't do those things because structurally or sequence-wise, they just lack the components or the sequences to be able to do those things. And I think we are going to find additional things in other parts of the genome that find functions that we haven't even hypothesized yet. There's 20% of the genome that's basically non-repeat, non-constrained DNA. It's mostly in introns. And it seems like it's junk. Is that 20% entirely junk? I'm willing to bet probably not. But we don't know yet. And I agree. I think we take a wait and see approach on that. Let's see. But for other parts of the genome, we can be really sure. We're really, really strong in terms of our understanding of a large part of the genome now. We don't have to wait and see on that stuff. We should certainly keep investigating it because a lot of it has been shown to have implications for human disease. So we should 100% keep seeing what's going on here. But like right now, given what we know in 2024, it is extremely reasonable to look at all that stuff. And it's not just assuming or extrapolating, and it's not a science stopper to say, hey, we've done the math, we've done the work, we've documented what these things are and what they do and don't do. That sure looks like junk right now. So here, if you want to do that, and then we can do our closings, and then we'll be.
Well, can I respond to that, or?
Please, yeah.
Yeah, I appreciate what you're saying, Dan, and I appreciate that you're trying to keep an open mind here. But it's not like I'm the only person who's suspecting mass functionality in the genome. I mean, we have people like you and Bernie and John Maddock who are saying that the genome is highly functional. And I just, this quote that I want to repeat from Maddock and Amaril's book, RNA, the Epicenter of Genetic Information. They said, while the story is still unfolding, we conclude that the genomes of humans and other organisms are not full of junk, but are rather highly compact information suites that are largely devoted to the specification of regulatory RNAs. And just to interlude, we already know that it is possible for just about every kind of genetic element in any degradatory state that you can find to potentially have a function. So I don't know of any specific example of this is degraded too much, or this can't have a function. There's so many examples of function at various states that we're not speculating anymore about whether this or that can or cannot have a function. It's more like, does this or that have a function? So they go on to say these RNAs drive the trajectories of differentiation, underpin brain function, convey transgenerational memory of experience, much of it contrary to conceptions of genetic programming and the dogmas of evolutionary theory. There are people out there, mainstream biologists, who do believe that the genome is largely functional, just like I believe is probably going to turn to, is indicated by the evidence we found so far. Now, we're in a place where both you and I acknowledge that there is still a lot of work left to be done in research, right? So we both know that it could turn out that I'm wrong. It could turn out that you're wrong. But what I see in the trend line of the research, again, coming at this from the perspective of 20 plus years ago, when people would say that junk DNA is an absolute defeater of, of intelligent design. And now you even said, that it's very possible that these things could turn out to have function in many cases. People weren't saying that 25 years ago. There's been a major shift in the thinking. And if people want to see the kind of major paradigm shift that's taken place, go to my blog post, again, that I mentioned on Evolution News. The title is Junk DNA from Three Perspectives, some 3 quotes. Look at the final category. It's full of papers, mainstream authorities saying things like, we used to think this is junk, but now we know it's not. Of course, they haven't studied every example of that type of element to confirm what every single one does. But what they're finding is that all these different classes of genetic elements can potentially be functional. And that to me means that the burden of proof is now on basically the evolutionist who's claiming that it is largely junk to show that it is junk, when we know that just about every class of element can, and in many cases is already known to have function.
So do we want to do like a brief closing thing, or do we want to do questions? How do you want to do this, Steve? I think probably.
We can get some questions. I mean, I'm
cool. Then we'll do just real brief wrap up.
That's cool.
Yeah. Mm-hmm. So, yeah. Yeah, so a lot of that is actually in very close proximity to genes. So we have these regulatory elements so basically a little bit of background. You have, sorry, don't ask a biologist to give a short answer ever. It's just never going to happen. So the real basic for everybody, structure of a gene, you have your region that codes for the protein. And then immediately upstream of that's before it in the sequence, you have your promoter. And that's where the enzyme that makes the RNA attaches. It starts at the promoter. Now, that promoter, you can inhibit that promoter, or you can upregulate that promoter and its affinity for the polymerase by layering other proteins on top of it. And the way that works is the DNA will actually, if you go further upstream of that gene, like 1,000 or 2,000 or 5,000 bases, you're going to have these shorter sequences called silencers and enhancers. And what happens is the DNA, I'm going to try to do this with my arms, it physically bends over. So if you have like a silencer over here, a protein grabs onto that piece of DNA and the DNA bends. There's another protein that bends the DNA. It bends over so that the protein on the silencer basically sits on top or adjacent to the promoter where the gene is going to start. And it's that range of bases in between that needs to be a certain length in order for that hairpin bend to work correctly. Basically, if it's too long or it's too short, you miss when you bend it over and you don't hit the promoter. So it's not that it's kind of a completely separate thing that, well, what about enhancers and silencers? It's that we can look around it at these very tightly constrained sequences that have to be fairly specific distances from each other. And you wouldn't put an enhancer in the middle of that region because it wouldn't physically reach the gene, right? It wouldn't match up to where it would be functional.
I was muted when I asked the question, but your answer was brilliant. But nobody heard the question, which is basically, I had asked him on the energetic non-coding DNA, which was the spatial DNA, even though it's mainly where it's at in the genome as opposed to the specified sequence of it, could that still have function as far as promoters, enhancers, and silencers? And that was the answer that he had.
If I could I say something on that, Steve? I've been looking into enhancers, and I find them really, you gave a great description of how they work, Dan. I appreciate that. I find them really fascinating because what they show is that DNA that is very, very far away from a gene, thousands of bases away from a gene, maybe in something that you would expect to have no function, some repetitive DNA element or whatever, can actually be involved in regulating that gene. And there's many enzymes and RNAs that actually are involved in, opening up the chromatin on parts of the DNA so that then they can be exposed far away from a gene and then, bringing parts of the genome together through these, what's called these topologically associated domains or TADs that bring together parts of the genome. The genome is literally a dynamically changing structure in its 3D structure during the course of a cell's development, differentiation, its cycle. And this allows different genes to be expressed in different ways. And so there is so much possibility for function here. And I'm not saying, therefore it's all functional. What I'm saying is that what we know about enhancers has really been a game changer in terms of opening up possibilities for what could or could not be influencing the regulation of a gene. Again, you could have repetitive DNA far, far away from a gene that is involved in regulating it under this enhancer model.
And the other question I had, if you did have a spurious translation of a transcription of something that was only like 1 FPKM, which is really, really low, how would that have any effect on the cell, whether it be upregulation, downregulation, whether it has any effect on methylation or histomodification, anything? How can one transcript have any effect on the cell if there's only one FPKM?
I assume you're asking me that question probably because I think Dan.
Okay, all right. Both of you, yeah. But,
I can give you an example of it would but I'll let Casey go if you want. But I can give you an example of how it would.
I'm not a geneticist, so I'm not going to have I'm definitely not going to have the depth of knowledge that Dan does. But I will say that, when you're talking about regulatory cascades and developmental gene regulatory networks, they often start with one gene that starts a cascade in that entire network. And then that network goes and does its thing. Okay. So presumably, one transcript or a very low number of transcripts is all that's needed to initiate that developmental gene regulatory network. Developmental gene regulatory networks are basically networks of genes that are involved in the development of multicellular organisms. And it's amazing. You can read some of the papers on these and they look like circuit diagrams where, this gene is interacting with that gene. This gene turns this thing off. If this is on, then this, regulates. It's just highly complex regulatory logic. It looks like something from an electrical engineering paper. It's literally like, electrical circuits is what they look like. But there's logic there. And you can start these things off with, and some of the papers that I cited earlier that I read talk about how regulatory proteins that are involved in these developmental gene regulatory networks can be active and functional at very low copy numbers. So I think this is a good example of that.
So that would be considered a functionality then.
These are vital. You when you tweak a DGRN, you basically, there's a lot of research that shows that it is highly, highly deleterious to the embryogenesis of an organism. And you can't really mess with them or they typically die.
So, I'll put down the spot real quick on that. So if that's the case, real quick, how would you ever know that single transcript didn't have function? How would you ever say that something's so degraded that it cannot have any function whatsoever? It cannot have any enzymatic function, nothing.
what Casey just described, those are protein coding RNAs though. So that's a transcript of a gene that then is translated to a protein, and then that protein does, so it's going to be like a kinase, like an enzyme that phosphorylates something else, and that leads to a cascade of kinases where they all phosphorylate each other and you amplify the signal. And so, but that's not the kind of, that's, we agree that's functional, but protein coding.
What about non-coding transcripts?
Right. So there's a way that you can have like long non-coding RNAs that are regulatory at small numbers, because they would act, rather than acting on a target RNA, they would act on the genome, right? So if you're targeting a specific place of the genome, we have diploid genomes, we only have 2 copies of everything. Now there's a lot of stuff that's kind of repetitive, but if you're talking about a specific gene sequencer or grabbing onto and inhibiting like a specific enhancer, for example, right? There's only so many copies of that thing in the genome. And then if you're talking about there's so much, we didn't even talk about like persistent methylation or anything, because that changes the number of targets in the genome. When your genome is all condensed up, then it's off the table, right? And that cuts both ways in terms of the arguments we're making, because one, if something's persistently methylated, it's not doing anything because heterochromatin, but also it decreases the threshold for targeting for other parts of the genome because there's just fewer targets, right? So then your threshold for how many regulatory whatever do you need to affect what's available to bind to goes down. And in that situation, Steve, you have, you take your fraction of the genome that's not condensed, right? So it's available to be acted upon, and you have a specific target, then an RNA transcript at very low levels could potentially grab onto that specific target and affect something.
But even if something is methylated, it still has the potential to be translated later on.
You would have to you'd have to undo it.
Undo it.
Yeah.
Right. Unmethylated.
Right. And after, and I will just say on methylation, just one tiny little detail is that after embryonic development, because your methylation pattern gets reset during embryonic development, post-development, a very large fraction of the human genome is persistently methylated. There's a lot of stuff that is involved in development and then not in anything else, and we just shut that stuff down.
It just stays methylated.
Which is not to say it's not functional, just to be clear, because development's really important, but just to make that clear in terms of the scales for everybody.
If I could chime in on that, I did cite earlier the Maddock and Amaral book, which noted that there are long non-coding RNAs that are present in very low copy number that are functional, and that's a form of non-coding DNA. And you're right, Dan, that I was talking about Hox genes. Obviously, that's an mRNA transcript. The Kellis et al. Quote, again, was saying that polyadenylated RNA, which is typically, talking about mRNA, had very low transcript levels. So, I think that this actually does address what Kellis et al. Said. But at the same time, you've got to initiate transcription. And somehow those genes, those protein coding genes are being initiated, presumably by some non-coding RNA. And so I think that there's room here to initiate, absolutely. This is
you don't need RNA to initiate transcription.
You can have RNAs that are involved in regulating the initiation of gene expression. That's very, very common. And so the idea that you can have non-coding RNAs in low copy number involved in creating, something that is just going to have a very low activity in the cell, I don't think that's at all a stretch.
Quick, I'm just going to knock these out real quick. They're not really that biological in nature, so it shouldn't take a long time to answer. Just from Wintry Night, he says, I would like to know how Dr. Dan responds to the quotation that I posted from Dan Grauer, who he cited, claiming that the ENCODE study conflicts with Darwinian predictions. I'm sure you're familiar with the quote.
I alluded to that earlier. That's the if ENCODE is right, then evolution is wrong thing.
Right.
And so that's the paper on that was from 2017, and that's been kind of refuted. And if you go to that paper now, if you go to that URL, it has a correction right at the top. Basically, Grauer says, hey, this math that you're about to read, this is wrong. Read this other paper instead. This math is correct. And the correct math removes the objection.
Gotcha.
And I will say Grauer was a reviewer on the later paper.
Go ahead.
This gets to the issue of something called mutational load. And it's the idea that you need a lot of the DNA in our genome to be junk, because otherwise you're going to be accumulating lots of mutations in very, important parts of the genome, and that will kill us off. And somewhere around, every generation, everybody has about 60 new mutations compared to their parents. And if every single nucleotide it has to be the exact, sequence that it's in, and you're getting those mutations, presumably then nobody would ever survive these 60 mutations. And so Dan Grauer says you have to have lots of junk in the genome to accommodate these mutations. I understand, Dan, that you are not making this argument tonight. And so, but I just want to say that there is good evidence. There was a paper that was co-authored by Ewens in Genome Biology and Evolution, who's a very big guy in the population genetics community. And it basically shows that you can have non-neutral mutations that are deleterious, but they don't kill you immediately. So the fitness effect need not be absolute. It could be relative to other individuals in the species to be able to not have this need to have all this basically junk that doesn't do anything whatsoever in order to get around the mutational load issue. You could have basically I think a lot of non-coding DNA that can accumulate some mutations, and those will have effects on this actually is why I believe so much non-coding DNA is associated with disease, because it does have functions, but only when you accumulate, certain mutations do you see that this non-coding genetic element is associated with a disease. You may have synergistic epistasis where eventually you get enough mutations that this non-coding genetic element is causing some disease in an individual. And so I think that it's an interesting high association of non-coding DNA and disease. It shows that the non-coding DNA and various diseases, it shows that it is functional, but it may that first initial mutation that it's receiving may not kill you. It may not even be all that deleterious, but eventually enough compound and it could do something negative. And these are all ways of getting around the mutational load issue.
I do just want to say on the disease thing, I think that's extraordinarily strong evidence that these sequences are non-functional, because a lot of what happens with these, it's going to be things like it's retrotransposons, it's going to be SVA transposons. These are active transposable elements in the genome, and our genomes work really hard to shut them down. So they're not doing their jumping gene activity. And when they mutate, they evade our kind of countermeasures and they mobilize. And that's been shown to be associated with cancers, with neurological disorders, developmental disorders. And so it's not the case that indicates that they're functional. It shows that they're actually harmful to you when they are biochemically active, and we do a lot of work in our cells to make sure they're not, right? That's what that data shows.
Or that their jumping function is not their function. They may be doing something else.
I'll save this last question for the very, very end, so you guys can do your closers here. I do have one other question, which you hadn't talked about in this particular debate, but it was brought up before, and I really want to get some clarity on it because I'm sure people will be asking about it at some time. But there was something brought up about the amount of energy that these wasted junk DNA would be for a transcription, as opposed to any kind of mechanistic or machinery that would be involved to eliminate these things as far as energy for fitness. Can either of you address that real quick?
Yeah. So this is one of the objections against the idea of junk DNA, because the idea is that junk DNA is basically energetically costly. So even if it's not like the molecules aren't harming you in any way, so you wouldn't select against like the presence of the RNAs, the fact that it's there is costing you energy that would confer fitness cost. So you would expect your genome to kind of purge it out over time. This has been addressed. There's actually a paper in 2015. It was who wrote this paper? It was, I have it right here, it was Lynch, 2015. The bioenergetic cost of a gene. And basically what this paper goes into is how the cost of transcription going from DNA to RNA is quite low. Translation going from RNA to polypeptide is very high. So what we see is for organisms that have relatively small effective population sizes, and mammals tend to be in that category, what you see is that the differences in fitness due to the presence or absence of junk DNA is below the drift threshold. So the cost is not high enough to select against, whereas the cost of spurious translation is sufficiently high that you can basically suppress translation in the RNA, but not the transcription. And then you could also look at things that have very efficient genomes, like bacteria. They're much smaller cells, so they're on a tighter energy budget. And they have huge population sizes, right? So now their ability to select for very fine differences in fitness is enhanced because of those population characteristics, which means that for bacteria, it's above the drift threshold, and they actually can suppress junk DNA in their genome. So in like E. coli, for example, it's all either coding sequences or regulatory sequences, very streamlined, because they can actually, based on their effective population size, efficiently select out any junk that might accumulate.
Let me say I actually agree with what you're saying here right now. There's been some research coming out of the ID community. We do research that has shown that actually mildly deleterious um, uh, traits, the natural selection is very inefficient at weeding those out, and that they can often accumulate in the genome. And so I think that if you're talking about a trait that is only mildly deleterious, it is going to be difficult in a large population of metazoans, for example, to weed those traits out. That being said, I'm not so sure about this argument that because it's not the case that the genome is transcribing everything in every cell, right? I mean, looking at ENCODE, one of the most interesting findings of ENCODE, in my opinion at least, the way I read ENCODE, is that Dan, you just reeled your politics, by the way, but it's okay. Yeah, your donkey cup there. Anyway, no worries. So the way I read ENCODE is that certain elements of the genome are active at certain times of a cell's cell cycle or the life cycle of an organism or certain cell types. If you have a bone cell, you're not going to activate the genes for encoding hydrochloric acid. You don't want to express hydrochloric acid in your bones, or there's many examples like this. So we're not transcribing, and we're not going to activate them through whatever non-coding DNA is regulating those genes. So I don't think that we would expect that under an ENCODE mass functionality view of the genome that we have to be transcribing everything in every cell all the time. So I don't see this as necessarily a big issue.
Yeah, those are both great answers, by the way. I'm glad that we got out of the way because that has been a question that has been brought up many times. So let's go ahead and wrap this up. I'm going to give you guys maybe 5 minutes for your closers. One quick question at the very end. But Casey, since you started, you want to do a quick recap of your position and things, and then we'll let Dr. Dan close us out. Is that cool?
Sure, that's fine. I'll do my best to keep this I didn't prepare a closing statement, so don't worry.
About your voice. Hey, look, the time constraint's not a big deal to me, but I know your time's valuable. Plus, your voice is kind of sounding pretty raspy.
I've got no more talks for the next week. So if I lose my voice, there's nothing left to do right now. So let's just kill it. So basically, my position is that the evidence that we've seen from thousands and thousands of papers that have discovered function for just about every conceivable non-coding DNA element in many different states, what Dan would call degraded, what Dan would call perhaps in its pristine form, we have found so much evidence of potentiality for function of non-coding genetic elements. And also we found evidence for mass transcription of the genome. This argument that Dan made that there is less than one transcript per cell, and that means that, quote, absolutely not can be functional. I think that we really did away with that argument, to be honest with you. I think we showed that ENCODE's methodology shows that it's doing an average. And just because some genes are not expressing a particular non-coding element, that doesn't mean that it's not highly active in other genes at other stages of the life cycle or different cell types. So I just don't find that this ENCODE low transcript level on average argument has in any way precluded function. Instead, what I've seen is the fact that we are transcribing a huge percentage of the genome. We already know that is prima facie evidence that it is doing something in certain cell types in certain stages of the life cycle. The reason why our genomes are not streamlined like E. coli is because E. coli only has one cell, all right? We are multicellular organisms. Our complexity is orders of magnitude greater than, now E. coli is pretty complex, but our complexity is orders of magnitude greater than E. coli. We have to have all kinds of different cell types, which are going to be differentiating during development of an organism. You're going to be creating blood cells, skin cells, bone cells, neural cells, and there's thousands of different types of cells in the human body. And it's the non-coding DNA that basically determines which genes get expressed in an organism or in a particular cell type. And that determining of the cell types determines what kind of a body plan you grow into. It literally determines why you are what you are. The reason why Steve, you, me, and Dan are not chickens is not because of our genes. I mean, our genes are like 75% similar to a chicken, all right? It's because of the non-coding DNA. The non-coding DNA is literally providing the blueprint that makes you a human being and not a chicken, because we are expressing these genes these protein coding genes in different ways. And that is the operating system of the genome. in the non-coding DNA that is basically regulating and controlling your development to make you who you are. And it's incredibly complex. So yeah, there's the reason why E. coli doesn't have all this junk DNA isn't just because it has, very efficient natural selection at work. It's because it doesn't need it. It doesn't need this junk to create all these different cell types and have, all these different variations of suites of genes that are being expressed in different cells. So what we found, I believe, is prima facie evidence from these thousands of papers, and especially ENCODE, which found basically mass transcription of functional, evidence of function throughout the genome, that this should change our default assumption that Dan has, I think, sort of articulated and exhibited for us many times tonight, that if we don't know what it's doing and it looks like it's not functional, then it probably is not functional. I think that assumption, and there's a lot of people out there who would agree with me. I found a lot of quotes, people saying, look, you can read that article I put on Evolution News, citing all these quotes, that we shouldn't assume that this is not functional anymore. And so I think that what we're seeing in biology is that a paradigm shift has taken place about junk DNA. And intelligent design predicted this paradigm shift. Evolution did not. Evolution instead has served as a science stopper. I mean, for goodness sakes, there was an article in the journal Science which said that the term junk DNA repelled. It said that it repelled mainstream researchers from studying non-coding genetic material for many years. So we actually have mainstream top scientific journals acknowledging that this idea of junk DNA, which came out of the evolutionary paradigm, has, quote, repelled mainstream researchers from studying non-coding genetic material. On the other hand, ID has encouraged researchers to study genetic material. You can go to our research homepage on our website, and we have researchers in the ID community who are actively studying function for junk DNA. I'm not going to out some of them because some of them are in very sensitive positions in their careers. They wouldn't want me talking about them, but it's just a reality that ID has inspired scientists to study function for junk DNA. I know quite a few people personally who that applies to. So I think what we're dealing with here, at the end of the day, a lot of where I think Dr. Dan's arguments came down are that he thinks that they're not functional, but we really don't know that. It's the same argument. It's the same dynamic of the conversation that we've been having for decades. And I have not seen any good evidence tonight from Dr. Dan that any particular genetic element that he cited cannot have evidence of function. So, but instead I've seen that the literature is replete with function, and the trend line of the research shows that we should anticipate more and more function is going to be found. And I think that these percentages of functional elements in the genome are going to go up, up, up. And we're just getting started. I mean, it could be another 100 years before we cross that 50% threshold, but I predict we're going to get there and we're going to go above that.
All right, Dr. Dan, go ahead and do a closer. Then I'll do a quick super chat, and then we'll wrap it up.
I just first thank you just at the very end there. I don't know if you meant to do that, but thank you for acknowledging that right now we've not crossed the 50% threshold. I appreciate
not
that.
from what we know. Not from what we know.
Okay.
Yeah.
Great. And that's far. I do want to say, no, when I want to start a debate, I just want to say that thank you for saying that, because that is at odds with what comes out of the anti-junk DNA community.
Well, no, there's evidence for function. No, let me say this. There's evidence for function. I think that ENCODE provides evidence for function, and our default assumption should be that it probably is functional. But we haven't specifically identified the function for the vast majority of the genetic element. I said that in my opening statement. This is nothing new. Everybody agrees with this. But what we found is a far cry from where we were 10 or 15 years ago. We have made huge strides towards seeing that the genome is mass functional. I think ENCODE provides good evidence of that.
I liked how Dr. Rana answered that question. Rana answered that question when you asked him. 51 to somewhere between 51 and 100%. Take your pick.
Right. Yeah. No, I do think that is at odds with much of the community, but we'll move on. I'll just kind of briefly summarize kind of from my perspective here. So what I've kind of tried to do with this conversation is really go through the nuts and bolts of what we know about the genome, what we have directly experimentally documented, right? So this is, contrary to what Casey's been saying, the arguments that I've been making are not based on ignorance. We're not saying we don't know. We're saying here are the functions. They're not doing and cannot do those particular functions. So in order to say those regions that we've been talking about, the lines, the other retrotransposons, pseudogenes, endogenous retroviruses, if we're going to say that those things are functional, we need to propose new functions, because we have abundant evidence for a very small fraction of function in those regions. We've got a handful of lines, for example, out of the 500,000 that there are, that we've documented function. we've got a handful of pseudogenes, a handful of endogenous retroviruses where we've kind of co-opted, we've exapted either the genes or the promoters. But for the rest, we know, not based on ignorance, not based on we haven't looked into it, but based on specific genomic analysis, we've shown that they are not doing the functions that we have directly documented in a small fraction of those elements. So while the function is widespread across all of these different elements, it is extremely rare within each element when you look at its frequency of retrotransposons, ERVs, whatever, right? That's the big takeaway. And it is based on known data, not we don't know, therefore assume it's junk. And I wish we could talk, everybody, for another hour, because there's so many things we didn't get into. We didn't talk, I just briefly mentioned persistent DNA methylation, how that affects things. We didn't talk at all about knockout experiments in mice that knock out millions of base pairs at a time from these regions with no cost. There's so many different things that, like, this is a conversation that, could take 3 hours, 4 hours, and we still won't touch on everything. But I think that's where I'll leave it, where basically for the parts that we talked about, right, these specific regions of the genome, lines, ERVs, pseudogenes, I think I presented specific numbers of specific data, not based on ignorance, but based on specific experimental results that show that for the functions that have been documented and proposed, most of those things are unable to do those functions based on their sequences and their level of activity. And that's, I think, where we can end it. And Steve, you said you had one more thing.
Yeah. I just had to unmute. So let me read the super chat. And by the way,
actually, sorry to interrupt. I should just say, based on those things, we can confidently say 8 to 10% of the genome is functional, and a much larger fraction, very confidently, 60 to 70% is non-functional, with probably like 20-ish% still kind of out there. That's kind of where I land on all this.
Dan, I hope that you and I both live long enough to be able to have this conversation again in maybe 20 or 30 or even 40 years. Because I think what we're going to find is that a lot of the functions that you thought had to be the function for these elements turned out to be other options of functions. So
I have to be clear.
Because history shows this has happened over and over in this debate.
This goes to this last
I should be clear, though.
Go ahead. Yeah.
Let me just be clear, though, on that, because I don't think they have to have those specific functions that we've been talking about, but those are the functions that have been documented and proposed. So if they are going to be functional, we need to come up with new functions for them.
Well, I cited functions for degraded repetitive DNA that is different from the function that you said it had to have. So I think we've already seen that it's been documented. But anyway, let's do it again in a few years.
We can do this again. I'm on the schedule for like 2030. Yeah.
I'm actually going to answer the last two for myself, if I may. One of them says, okay, so this is Blessed Be. He says, why can't scientists admit that they are wrong about junk DNA? And the real simple answer to that is, if they are wrong about something, scientists will admit to it based upon the evidence. That's, I think, everybody will agree upon. But this last super chat I want to read, and I'm going to address it myself. It's from Rich Torres, MMA, and he says, Christians stop arguing science with unbelievers. The worldview is fundamentally at odds to the scientific method. And let me address that, because science adheres to methodological naturalism. And as Stephen Jay Gould said, there's a non-overlapping magisterium between science, philosophy, and theology, in which case there are different domains of discourse. And so I actually encourage Christians to talk about science. I think it's amazing when they're able to reconcile their theological beliefs with the scientific information that's out there. I think that we should encourage that rather than discourage it. That's my personal position. And I actually appreciate Dr. Luskin coming on doing that as far as an intelligent design advocate. I am obviously not an intelligent design advocate, but I think it's impressive that they're at least taking a scientific methodological approach to it. And I think probably even Dr. Dan would agree upon that. But I don't want to put words in his mouth. But I think we all agree that there is a completely distinct domain of discourse between the theological aspect and the biological aspects here.
Steve, I would, in response to that questioner, I would say that actually, and you can look at the quotes I posted, a lot of scientists have acknowledged that they were wrong about junk DNA. And I want to close with this will be the last thing I'll say. I want to give a quote just from Francis Collins, who of course is a Christian and an evolutionist. He's a very well-known, advocate of evolution. He's also head of the NIH. This is what he said in 2015. He said, in terms of junk DNA, and he was an advocate of junk DNA years and years before this. He said, in terms of junk DNA, we don't use that term anymore because I think it was pretty much a case of hubris to imagine that we would dispense with any part of the genome if we knew enough to say it wasn't functional. there will be parts of the genome that, are just random collections of repeats like ALUs, but most of the genome that we used to think was there for spacer turns out to be doing stuff, and most of that stuff is about regulation. So maybe, Dan, we each get a little bit from that quote there, but I think it's a good quote to think about.
Well, I want to thank both of you for joining me. Like I said, I know this was a work in progress to make happen, and I appreciate the effort that was involved in making it so. And I'm sure there's going to be a lot of videos talked about this, because I think both of you gentlemen answered these questions up front and very well. So I want to thank both of you. If you have any questions, please leave it in the comment section. I have all the links in the video description, and I'm sure that if you have something directly to get to either one of them, I will forward on the messages. And with that, I will bid you guys good night. You guys know my plans for tonight. You two here know what my plans are for tonight. Not fun.
Good luck.
I have. Yeah, thank you. So with that, thank you for my roommate's laughing on that one. Thank you for watching the Non-Sequitur Show, and we'll see you later. Good night.
Thank you, guys. Thank you so much.
Thanks, everybody. So that was my debate with Dr. Casey Luskin from Discovery Institute on the topic of junk DNA. The thing that I want people to take away from this, and I've highlighted this in a separate short video, which I've linked down below, the thing I want people to take away is what Dr. Luskin said right at the end there, where he acknowledges that based on what we know right now, we cannot assign function to more than 50% of the genome. The actual number is way less than that, but the threshold that I had asked him about was 50%, and he said, based on what we currently know, we can assign function to less than 50% of the genome currently. And on that, I agree. And I think where we disagree then, it turns out, is what's going to happen in the future as we keep digging into the rest of the genome. And I think the answer is clear. Obviously, Dr. Luskin, anti-junk DNA people, and a lot of intelligent design creationists disagree with me. And that's okay. We're going to see what the data show, but we already have a lot of that data, as I talked about, and the human genome is largely junk. Thank you for watching. If you enjoyed this, please remember to hit like, subscribe to the channel, leave a comment, share this video. You can become a channel member. Thank you, Ian and Charles, for your generosity. Those are my second-tier members as I record this. And as always, don't get fooled.